about the video, but in any event, I can imagine the video because it talks about innovation and caring and our goals. I also want to thank our partner in today, Pisces. It's been great partnering with you. And thank you all for joining in person. 30 years ago, Friends of Cancer Research was founded on a simple idea. Science supported by the right policies can save lives. Let's remember that nothing gets to the patient without FDA, and it's extraordinarily important that continue the innovation. The idea feels more urgent today than ever because we really have the science. When Friends of Cancer Research was started 30 years ago, we were treating cancer by organ sites. The idea of personalized medicine was just a concept. The idea that we could really intervene early wasn't there. So there are just so much that has happened, and we're thrilled about it, but there's much more that has to happen. Cell and gene therapies are changing what is possible. Again, 30 years ago, that was not possible. Patients who had no options now do. That is why we are all working. That's the point of innovation and the point of science. But progress does not happen on its own. It depends on strong regulatory funding and policy framework. It's more than funding. Funding is important. Science is important, but there is a mindset that has to happen at FDA of innovation and understanding patients' needs and where we need to go and what will benefit the patient. It's never perfect, and those frameworks are under real pressure today. This is why we're convening this meeting today, bringing together researchers, regulators, industry, and patient advocates. Science needs champions. Everyone in this room is a champion, and we know why we're doing this work. It's for patients. Patients need us to show up. Their voice must be heard. We are grateful to the Parker Institute, started belief in, we started this collaboration, and urgency drives this work. Thank you for being here, and we look forward to an important meeting, and thanks all those who helped participate, and I expect a very meaningful outcome. Oh, I forgot I'm supposed to introduce Mark Stewart. I don't know how anyone can miss him, but anyway, but you can't miss him. But Mark Stewart, thank you for being here, and thank you for taking over.
Lift those up a bit. So thank you, Ellen, and thank you all for joining us, both in person and virtually, for today's meeting, Unlocking Next Generation Therapies. We're really glad to be partnering, as Ellen said, with the Parker Institute for Cancer Immunotherapy on this. It's been a strong collaboration over the past several years and has helped continue to move this conversation forward in a meaningful way. Before we get into the content today, just a few bit of housekeeping items. Everyone registered for the meeting today should have received an issue brief this past week in advance, which outlines key themes we'll be discussing today. Additionally, you can access it through the executive summary in the booklet on your table. Additionally, you'll see a QR code in the agenda that links to Slido. So that will be how both in-person and virtual attendees will submit questions throughout the meeting. And so for each of the three sessions today, we will include time for audience Q&A towards the end. So we encourage you to submit those questions as they come up throughout the meeting. With that, I would like to transition to a few framing remarks to help set the stage for today's discussion. But before that, I would like to have the panel one come to the stage so we can have a seamless transition into the panel discussion. And so while they're making their way to the stage and hopefully in the correct seats, I will start by just giving a brief overview in terms of what we've been hearing through conversations across various stakeholders and where we think there's real opportunity to really make progress. So today's discussion, I think, is really about how we translate momentum and the science and the desire to move quickly but safely into operationally feasible and efficient development pathways. And so to ground us, just a quick overview of what we mean when we talk about cell and gene therapies. These are treatments that involve using or modifying a patient's cells or introducing genetic material to treat, in some cases, potentially, and in some cases, potentially cure a disease or cancer. And so they can fall into three broad categories: cell therapies, which use living cells from the patient or a donor; gene therapies, which alter genetic material within a patient's cell; and cell-based gene therapies, which combine both. And so on the right, you can see a simple paradigm of CAR T therapy in which T cells are isolated from the patient. They're engineered to target a cancer cell, expanded, and then delivered back. And so this paradigm, as Ellen noted in her opening, has produced real breakthroughs, but it's also a useful illustration of why these therapies present such distinct challenges from traditional therapies. They're highly personalized, they're biologically complex, and that complexity shows up across every stage of development. And so that really brings us to the core of the conversation that we hope to have today. Cell and gene therapies have characteristics that don't always map neatly onto frameworks originally built for small molecules or even conventional biologics. Many are manufactured on a patient-by-patient basis. Clinical trial designs are driven by small patient populations and product-specific considerations. And then you have manufacturing and delivery that introduce another set of operational complexity. And so these can obviously directly shape how development programs are designed and how they and how regulatory expectations are applied. But with that, I do think there's meaningful progress, and certainly progress that has occurred over the past couple of years. FDA has been engaged on these issues. We've seen a number of recent guidances and initiatives that reflect real openness to adapting existing frameworks for these products, highlighted by just a few select guidances here that I had to update last night with the release of a new one. But we have the 2023 guidance on manufacturing changes and comparability, 2025 guidance on innovative clinical trial designs for small molecules, the most recent guidance on plausible mechanism framework, and then just yesterday, one on CMC flexibilities for cell and gene therapies. And so in our conversations, it's not necessarily that there's a lack of flexibility per se, but it's really what happens between guidance and practice and how one interprets and operationalizes the content of these guidance documents. And that variability shows up in a few ways. You can have drug developers that are going through repeated iterations on protocol elements, drug developers taking conservative approaches to study designs and data generation, and together these are often driven by uncertainty of, based on real or perceived expectations during the development process. And this has real important downstream implications in terms of how quickly trials are able to start, how efficiently programs move forward, and ultimately how quickly patients can access these therapies. And so I do believe there's an opportunity here, because in many cases we're not necessarily needing to rely on new policy, but more so how to improve the clarity and consistency in how existing flexibility is used. And so where do we go from here? The issue brief, as I noted, we've shared ahead of today's meetings, organized around 3 areas where we think targeted action could make meaningful difference. And those map or coordinate with today's discussions as well. First, we have early phase clinical development. There's an opportunity here to better distinguish, we believe, where common approaches could be leveraged versus where true product-specific tailoring may be needed. I think this also ties nicely with ongoing efforts to leverage AI to accelerate early phase, and obviously to the extent that there's greater harmonization across documents certainly can benefit that. And so as a start, more structured starting points for protocol design and clear expectations during IND preparations. Second, CMC manufacturing. Here the focus, again, is on how to operationalize risk-based approaches across the life cycle and how that flexibility might be built in from the start, allowing evolution to occur within certain parameters rather than negotiated at each step. And third, clinical trial designs. As these therapies move into earlier lines of treatment, there are real questions around how one goes about selecting the appropriate comparator, the feasibility of randomized designs, and when randomized evidence is truly required or needed, and obviously the role of novel endpoints to make these trials more feasible. And so across all these three areas, the goal isn't to reduce the rigor, but to improve the predictability and efficiencies and focus effort on questions that matter most. And so today, through the various conversations that we've lined up, we hope to pressure test several ideas that are in the issue brief, hear from experts across the field. Areas that will come through in our discussions today will include elements of early phase development where there may be opportunity to have more consistency. What would a life cycle-based approach to CMC actually look like in practice? Where are there opportunities to rethink trial designs in a way that reflects the realities of these therapies? And what policy opportunities can set us on a path to ensure continued U.S. innovation in this space? And so I just want to conclude by acknowledging the working group members who contributed to this issue brief and to the thoughts that will come through in today's discussions. It certainly helped in terms of the breadth of expertise that was represented in this group, including clinical development, manufacturing, regulatory science, and patient advocacy. And so I also want to acknowledge FDA's continued willingness to engage on these issues, which is obviously critical in moving these topics and issues forward. So with that, I'd like to hand things over to our first session, which will be moderated by Lola Fascio and Ajay to moderate our conversation on early phase clinical development.
So good morning. Thank you, Mark, for setting us up really well there with this anchoring presentation. I want to thank Friends of Cancer Research and the Parker Institute for convening this meeting. And the working group we've had really insightful conversations, discussions with really very interesting ideas. As Mark mentioned, we're going to be focusing on how we can modernize early phase clinical development approaches and also discuss briefly potential solutions for streamlining, improving efficiency in late phase development. I'm Lola Fascio Ajay. I'm a medical oncologist by training, regulatory scientist with over a decade experience at the FDA in both the Center for Drugs Evaluation and Research and the Center for Biologics. I'm currently Vice President for Regulatory Science and Strategy at AstraZeneca. Started 2 days ago. I'm pleased to be here. I want to introduce my esteemed panel and again, thank all of the members of the working group for their contributions to what I expect will be a really great discussion today. So to my immediate left is Stephen Fleischer. He's a doctor of veterinary medicine with over 20 years experience at the US FDA, initially joining as a pharmacology, toxicology, reviewer of cell, tissue, and gene therapy products in CBER. He's currently the acting director of the Office of Pharmacology Toxicology in the Office of Therapeutic Products, OTP, at the Center for Biologics Evaluation and Research, CBER. Welcome, Stephen. Next is Dr. Vijay Kumar, who is a board-certified nephrologist who joined the FDA in 2020 as a clinical reviewer, became a clinical team leader, and is currently the acting director of OTP in CBER. Welcome, Vijay. Next is Travis Quigley, who is a leader in clinical development with more than 25 years of experience advancing innovative therapeutic programs in biotech and pharma. And he is currently Chief Development Officer at Colonia Therapeutics. Welcome, Travis. Next is Dr. Rosanna Ricafort, who is a pediatric hematologist oncologist and stem cell transplant physician with extensive experience in academia and is currently Senior Vice President, Head of Cell Therapy Medical at Bristol Myers Squibb. Welcome. Next is Dr. Nirali Shah, who is a pediatric oncologist, physician scientist, and her research focuses on the development of targeted immunotherapy approaches to treat high-risk blood cancers such as leukemia and lymphoma in children, adolescent, and young adults. And she is currently a senior investigator and head of the hematologic malignancy section of the pediatric oncology branch at the National Cancer Institute. And our raison d'etre, Chris White, is a patient advocate, a survivor of stage 4 mucosal melanoma, and he has contributed extensively to drug development in cellular therapy, and we're very pleased that you're joining us today, Chris. All right, so as Mark mentioned, really our task here is to talk about what are challenging areas or areas of pain points in early stage clinical development. And really what's underlying this is there are a lot of conversations that are ongoing around, the concern that the US will lose its lead as a research ecosystem where innovation is occurring. And so this conversation is not about, other countries and their increased contribution to innovative therapy development. This is really a very US-centric conversation to address areas that are challenging or not really addressing our current needs and keeping pace with innovation because of our very entrenched research ecosystem in cellular gene tissue therapy specifically, and not having the agility to really keep pace again with innovation. So I want to get right into the questions that we discussed. So I want to start with thinking about how early phase development looks like from all of your perspectives, how it's practiced today, where we run into friction, and how this influences how you conduct early phase studies. So I will start with Travis. From your experience, if you could touch upon what factors impact a company's decision regarding when and where, whether it's in the US or outside the US, to initiate early phase cell therapy trials.
Sure. So I think it depends on the company, of course, and how they think about developing their program. I think there's obviously concerns about speed and time. There's concerns about cost. We've seen that a cost of a trial in Australia is probably about half the cost in the U.S. Now, that's not a regulatory issue. That's more contracting with clinical sites and the cost of doing business. But then there's also, I think there's this perception or concern about, what's FDA going to say at an IND? Is it going to slow us down? We had a fantastic pre-IND meeting with the agency with great alignment, so we didn't have concerns there. The driving force for us in development to start in Australia was really the patient population. Given the development of CAR T cells for myeloma and the availability in the United States, along with T cell engagers, we really wanted to benchmark our program in a patient population that didn't have access and didn't have prior BCMA therapies. So that was really the driver for us to start in an ex-US setting.
Right, great. And Nirali, if you want to jump into the conversation, can you talk about any challenges that you encounter initiating cell therapy trials in pediatric populations, including specific examples such as aligning on dosing strategies and DLT definitions, work that I know that you're very passionate about?
So first I'll start off by saying what a wonderful way to have this meeting. I think it shows you how far we've come in cell therapy. The first CAR T cell approval was in pediatric patients, and I think since that time we've really come a long way. I think from an academic standpoint, there's probably two main considerations that tend to be a little bit of a pain point. One is that as a pediatric oncologist, you want to focus on the patients who have that disease at the time so that you can get them to the trials, and we often do have the issue of having to treat older patients before getting to younger ones. I think now we're in an era where CAR T cells have demonstrated clearly time and time again that they do have that potential of direct benefit. And we can probably start to scale back from that framework of it always has to be a couple of 18-year-olds or 16-year-olds. Because what ends up happening is that there are patients then who cannot access the study even though they are as much in need as an older patient. And you may end up slowing down the trial enrollment by having these arbitrary cutoffs. And some of them aren't super arbitrary, right? You have to 18 and age of consent. But you have to remember the patients who come to phase 1 trials have had that same prior therapy and are coming for a reason. I think the other issue that we struggle with a little bit in academia is that we are talking about very, very rare populations, especially in the relapsed refractory setting. If you have multiple trials that are happening at the same time, you end up having you struggle with issues of accrual. When you try to do multi-center trials, I will say that funding is a challenge. Some of the academic studies that we do, they are fully academia funded in some way through a series of grants, and that becomes challenging, and it's quite different from adult multi-center clinical trials, and I think we're trying to figure out what's the path forward. I would love to see that as we are doing more cell therapy and gene therapy in pediatrics, that pharma has, that there's less of a reluctance to engage in pediatrics first and not have to wait for adult studies to be started.
That's great. A little bit out of order, Vijay, I want to sort of follow up on what Nirali just mentioned, because there was a recent FDA CBER pediatric cell therapy workshop. Any thoughts around what's next at FDA just based on that really very rich discussion that occurred?
Yeah, thank you very much for including us in this conversation. We are pleased to be here. Yes, as all of you are aware, the framework for earlier inclusion of the pediatric patients in clinical trials is evolving. We held the first ever pediatric cell and gene therapy workshop last month. There was a robust discussion from thought leaders, both from academia and the industry. This feedback was particularly valuable for our office as we are moving towards a more disease context sensitive, mechanism aware framework for pediatric eligibility, one that does not solely rely on temporal sequencing of the studies from the adults. But instead, we ask the question, is early inclusion scientifically and ethically justified based on our current understanding of the disease, characterization of the product, and the mechanism of therapy? The other kind of big change is that the current stance in our office is that the timing of the pediatric enrollment should be addressed as a central design question, not as an afterthought to be resolved in the post-approval setting as a PMR kind of like, requirement.
That's great. That's
Thank
great.
you.
Thanks, Vijay. And just to thank you for that. That's wonderful. There has been a lot of discussion about growing trend of early phase studies being carried out outside the U.S. What's your perspective on that? Any concerns on this trend? Any downsides, upsides that you see with that?
Okay, so I'll try to, it's a hot topic, so I'll try to kind of like, do my best to kind of like, cover all aspects of it. Yes, we acknowledge that early phase studies conducted outside US are driven in a large part by operational considerations related to cost, faster enrollment of a diverse population, and the intent of regulatory convergence. From a regulatory perspective, we have a host of concerns on several key areas related to product quality, interpretability of scientific data, on safety and efficacy. So I'll just briefly kind of like, run through some of these. We have manufacturing concerns on, we have a adherence to risk-based, phase-appropriate CGMP, manufacturing chain integrity, facility inspectability. We have concerns on adequacy of the non-clinical safety package, particularly toxicology studies. We have concerns on good clinical practice and data integrity with verifiable efficacy data, study oversight for safety with local ethics standards and investigator accountability is another concern. We have concern on the informed consent process, specifically discussion of alternate therapies, communication of novel and unfamiliar risks, and commitment to long-term follow-up. But most importantly, we also have concerns about the applicability of the ex-US data to the US population, not just based on the demographics, but also baseline characteristics, definitions of refractoriness, and availability of standard of care therapies. The reduced US site experience in early phase studies can also delay the US experience in the phase 3 site initiations. As some of you are kind of like, aware, there is a big modernization that's currently underway in the FDA. We have a slew of initiatives and pilots, whether it is in affecting every discipline, whether it is CNPV, plausible framework, NAM, CMC flexibilities, single pivotal study as default, real-time clinical trials, unified platform for analyzing adverse events. So I'm just mentioning a few names here.
Yeah.
So when we look at it, if the companies are engaging with consultants and CROs that are not up to date on all of these kind of like modernization efforts, the sponsors may do studies outside the US. There may be a short-term kind of like benefit, but in the long run, it might prove counterproductive to them.
Thank you, Vijay. Nice plug there for the CROs. I'm sure they're very happy to hear that. So turning over to Chris, you've navigated, early phase study of TIL therapy on the patient side, so you have that experience. Can you tell us a little bit about what friction points you experience or that you are aware of that patients experience, and what's at stake for you in terms of where and how quickly a trial is available? Just speak from the patient perspective on this.
Yeah, absolutely. I am very passionate about this topic because I tell everybody I did not learn about the trial I eventually became a part of until I ran out of options of where I was at. And since I had a very rare subset, I was seeing a specialist. And when I got there, they told me about my disease and how rare it was and what they had available, but they didn't tell me what was available, elsewhere. They were only explaining to me what they had at their academic center. if there was more, education, patient education that like, hey, here's what you have. It might not be available for you now, or you might not qualify for it now, but, just to let you know that they're out there, and if the time comes, at least, you can get a little bit of a head start. I participated in a global trial which had 57 locations, and half of them in the US, but not a single one of them that I joined was in the state that I was in, so I had to travel out of state, and it was a centralized, location for them to grow my cells, so there was all that travel. So the patient awareness factor is huge, because if you were to bring it up to a patient and go, hey, here's a one-time treatment, you don't qualify for it now, but when and if you do, you can go try to do that, and then if that doesn't work, you can come back. But the risk versus reward factor of all the toxicity, that comes over time, and then, you're doing it as a last option. I explain to people, I'm the exception, not the rule. I was extremely heavily pre-treated by the time I got to it, but I would have jumped at it as soon as I qualified if I would have been approached by that, but the rules out there regulatory-wise say that they were not allowed to tell me because it was a conflict of interest, and so everybody at the FDA, really like to change those rules.
Yeah, and that's really a really good perspective there to provide. I think there's often this expectation that if a first-in-human study must commence in a relapse refractory population that has no other therapies available. And I think in cellular therapy, I think those of us who are working in this space know that that comes at a cost to even the type of a product. Your starting materials are really better perform better in a least or limited treated population. And so the points you're raising there around access are really important. So I want to switch gears a little bit and talk about some observations that sponsors have about certain protocol elements like dose escalation, design, toxicity monitoring windows, enrollment staggering, that these are revisited, iteratively across programs with very similar risk profiles where we have approved products, even in some cases, where the safety profile has been quite well characterized. So I want to talk a little bit about, your experiences, where this trend tends to arise and what drives it. So starting with Stephen, can you speak to where there may be some phase-appropriate flexibility from the FDA standpoint on the non-clinical package, and well-established versus where it tends to require more discussion, including how emerging frameworks like NAMS guidance and can inform the expectations that the agency has for sponsors?
Yeah, thank you. Yeah. We fully support incorporating flexibilities into the non-clinical program. And there's two that are worth highlighting today, and those are new approach methodologies, or NAMS, and leveraging. So we fully encourage the use of NAMS for the non-clinical program, and we have a common goal to reduce animal use. First, the recent guidance came out as a CEDAR guidance on NAMS, and it outlines general validation principles intended to enhance interpretability and reliability of data for regulatory decision-making. But I just want to highlight that guidance is focused on validation, and we fully agree with those principles, and they also apply to CBER products as well. However, I'd like to point out that NAMS do not need to be fully validated to support an IND, but sponsors should justify the appropriateness of that approach for the context of use and address whether bridging studies are necessary, and interpret the results within the context of prior knowledge and the broader clinical and non-clinical data package. But you are correct, there are differences in flexibilities of approaches. Alternative approaches are more established for certain products, such as CAR T cells that are transduced ex vivo for oncology indications. Most of that non-clinical work can be done in vitro. But there are products, such as novel mechanisms or first-in-class, where additional discussions should be had, and we invite you to come in to have those conversations. Examples would be CAR T cells that are with in vivo gene editing, such as with lipid nanoparticles, We would need to have more discussions about that, and often we're going to have additional studies regarding distribution or toxicology, and also recommending integration analysis. But non-clinical approaches are product-specific, and we recommend that you consider that as you're putting together the existing evidence while you're putting together your non-clinical package. And the other flexibility I'd like to discuss is leveraging, because I think it has a lot of ability to save doing studies. And that's the use of existing knowledge where feasible. And that knowledge could be widely available, could come from peer-reviewed published literature, and sometimes that knowledge is proprietary and may require permission or right of reference to other files. And while the ability to leverage does depend on similarities in the products, the big impact is it could reduce the need for new studies and allow to get to trials faster. So one example would be leveraging clinical data from CAR T cells in lieu of doing a non-clinical dose finding studies if the products are similar. So by using leveraging of similar products, you can use that to support efficacy and safety of the proposed starting dose, again, in lieu of conducting new non-clinical studies. And ultimately, both of those come down to optimizing the non-clinical program. So we recommend to optimize the program through early interactions with FDA. We invite you to come in and have meetings with us. And you should consider, when you're putting that package together, what studies are necessary, what flexibilities you want to incorporate into that non-clinical program, and include those justifications for that. And then we can have that conversation, agree to those designs, and hopefully with the goal of reducing unnecessary or repeated studies.
Thanks, Stephen. I know that some of what you mentioned has been described in some guidances that exist. I think sometimes a challenge that sponsors encounter is that when a new guidance comes out on the topic and does not include either a link to or a citation or a reference back to an existing guidance, I think there's concern that the new guidance is not relevant to cell therapy. For example, this one that came out that did not have CBER listed there. And so I'm wondering whether in the finalization process of that guidance, you might reference that many of those principles are applicable to cell therapies as well, and potentially refer readers back to an existing guidance. Because there's a lot of information across many, many guidances. And I think, as Mark mentioned, one of the challenges that sponsors encounters, particularly smaller companies, is knowing where to find the information. And coming to talk to you is always great, but I think it's even better when that information is accessible to a wide stakeholder community, including investors, including sponsors, who can really see the regulatory pathway very clearly.
Thank you.
So Vijay, I want to come to you, and can you talk a little bit about where discussion typically arises in early stage review and how the agency thinks about the distinction between early and late stage flexibilities?
In general, during the early stage studies, there's a lot more flexibility when it comes to several design features. We have the phase-based CGMP requirements, adaptive dose escalation, smaller sample size, exploration of biomarkers and novel endpoints, because the primary goal in the early phase studies is establishment of safety and dose response definition, and not necessarily drawing inferences on efficacy. The early, we generally encourage during the early phase studies to explore patient selection enrichment strategies there. In the late phase studies, we continue to offer flexibilities on adaptive designs, small sample size, Bayesian approach to analysis, of course, use of surrogate biomarkers to support approval. As many of you are aware, as of February this year, our default posture is to favor a single, adequate, and well-controlled study for, with confirmatory evidence. I must stress that at the FDA, we are focused on the quality and the persuasiveness of the clinical data, not merely on the quantity of the studies. We continue to be flexible in balancing pre and post-approval data requirements. We are open to extrapolation of indication to pediatric population based on the understanding of the disease, characterization of the product, and the applicable clinical data.
Great. Rosanna, do you want to, actually, let me go to Travis. Can you talk a little bit about navigating IND enabling and protocol design expectations as a biotech really quickly?
I think one of the challenges is just consistency and flexibility. So at this point with CAR T therapy, I think maybe there's room to kind of align on a DLT criteria, sentinel dosing strategy, and study pausing rules that are consistently applied across the board. And you have confidence in that going into an IND that those types of things in study design aren't going to change. I think the other piece that I think is important to keep in mind for phase 1 studies, especially where you have a safety review committee or a DSMB, is not requiring pre-specification of every detail in the study and allowing for some flexibility for a safety review committee to go above or below a pre-specified dose range or to implement prophylactic therapies prior to treatment, within a framework that can be provided in a protocol, but not necessarily pre-specified, so you can react to data and not need to go through a cumbersome and time-consuming amendment process. So just encourage more openness to the flexibility around some of those things.
Yeah.
Can I respond to the DLT question?
Sure.
Okay. So yes, DLT is one of my favorite topics because that's something that we wrestle routinely in our office. There are a diverse product range across CAR Ts. There is a diversity of indications, there's a diversity of sponsors, and our reviewers find the DLT very, very challenging. So let me give you an example to illustrate, and I would like the panel to say how they would resolve this issue. So we have a DLT based on a neutrophil count. Company A says the neutrophil count below a certain threshold is a DLT. Company B says, we don't believe in arbitrary thresholds. We look at, they go leukapheresis, we look at a percentage reduction for a DLT. Company C says, neither of those things are clinically meaningful. We only do DLTs for febrile neutropenia. And company D says, we don't even want neutropenia as a DLT because it's readily treatable. So how would you resolve when you have 4 companies and 1 reviewer trying to harmonize all of these DLTs? That's the challenge that we face in OTP.
Yes.
So maybe I could respond to that because I think, as a large sponsor, we have amassed an extensive amount of clinical data through our portfolio in myeloma, lymphoma, autoimmune disease, and there's tremendous amount of precedent, clinical and regulatory precedent that we learn from executing those trials. And so we have built internally, organically, our own common framework for how we design early development studies. But that's, we have the luxury of having that portfolio, whereas smaller sponsors do not. And so what I think would be helpful if we're going towards a solution-oriented path is having an intentional common framework that has structural starting points on those essential protocol elements that are common throughout all of early phase development in cell therapies, like DLT criteria, like safety monitoring, inpatient monitoring, like dose escalation, and how we proceed with that. And I think that would go a long way for elevating that. You pointed to peer review literature. We're not all going to do that, and it's hard for different sponsors across the world to really reference those. But if we had an intentionally designed common framework, that would be beneficial and really improve efficiencies in the pre-IND to IND space.
Yeah, I think recognizing that FDA is reacting to proposals often, and that those proposals have a lot of variability. And, in many cases, FDA is trying not to be prescriptive about how to move forward. But I think I take your point, and we had this discussion that having a common starting point where deviations are really rare and very specific to a very specific issue, whether it's lack of data or novel therapy where we expect, the clinical study to be sensitive enough to detect it. Nirali, did you want to add to this conversation on DLTs before we?
I was going to piggyback on that. I think you need that foundational framework because the four scenarios that you presented, they may all be right for the protocol, for the patient population, and for the study. Right, and so I always think about DLTs in the context of what you have for that. But again, I think what we're trying to say is that if you had a framework, you build upon that to say, neutropenia is important. Navigate what is going to be a DLT in your protocol, in your patient population. And so that's one checkbox. You can say, okay, we have to account for this, right? We had the issue with AML targeting. When you are targeting hematopoietic stem cells, and we expect that they will have aplasia. In fact, we account for them to have aplasia. And so that also cannot, your expected toxicity cannot also be your DLT. But again, so what we did in the AML, pediatric and adult communities to actually put out a working group and to say, we believe that these are common elements as a starting point for a foundation for DLT, for early phase, first in human, CAR T cell trials, and you build off of that.
Vijay, is that something that the agency OTP would be open to participating in some of this? Because you don't want this sort of ready-made and presented to you as a fait accompli. You probably want to be included and involved in some of these work
we
streams.
definitely want to be kind of like, because there is a tremendous amount of variability between the companies and the indications, as we have talked about. Any way that we can standardize, just like we have standardized the CRS and the ICANS criteria and the treatment, if we can standardize more of those features, that would be better for everybody, from the patients, from the sponsors, to the regulators, everybody.
Yes, that would be amazing. Okay, great. So I wanted to move the discussion to, I think we have a potential path to a solution here in the DLT framework, but sort of talking about approaches that could meaningfully reduce burden in the pre-IND to IND process to efficiently launch early phase studies from your perspective. So I want the panel to comment. So Nirali, maybe you can just sort of close the loop on the discussion around DLT frameworks and requirements for early phase studies, exploratory studies in particular.
Yeah, so I think that we can learn a lot from the diseases we've already treated. And so I think iterative development is really, really important. We can decide what studies were relevant in the first B-ALL, multiple myeloma studies, to sort of say we're going to streamline and not do all of these exploratory studies, because quite frankly, we may not have the funding to do so, especially with like smaller academic studies, and say we're really going to hone in on these. And I think that has to be a discussion to say we really don't think that this is necessary, feasible, or informative based on everything that we've learned over this decade of experience. However, we would like to do these other things. I think it was Travis who brought up the point about having a little bit of flexibility where you don't have to pre-specify everything, because you are learning, right? Protocols are living components. You have patients that are coming in. You are learning with the patients as you're doing the trial. And so you may decide after a couple of patients that we need to do preemptive cytokine release syndrome monitoring, because that is going to enhance the safety profile. And so building in some of those strategies to say, like, this is now an acceptable strategy in optimizing patient care. And so I think allowing for the prior experience to then inform your current and future studies will help to expedite the process.
So what I'm hearing you say, Nirali, is that, sort of customizing the requirements to the purpose of the clinical study and the context within which the clinical study is being conducted. And I think some of that will require some flexibility from the agency, but some of that may require some authorities, new authorities given to the FDA, so above the FDA. So I want to make sure to identify potential stakeholders for some of these solutions. A way to sort of differentiate kind of a proof of concept study and the requirements therein from a study that's really being commenced with the purpose of moving towards a licensed product. Okay, great. So Rosanna, can you provide a perspective on what types of solutions would actually reduce burden in practice? So not limited to protocol standardization. So could you include perhaps maybe rapid feedback mechanisms and such things?
I think we've already talked about a common framework, and what we've heard from Nirali and Travis is that we learn from every single patient that we treat. And sometimes we have data that changes or informs our early development design. And I think having that rapid ad hoc feedback, a mechanism for that with the agency would be helpful, something that's not burdensome for either the agency or the sponsor, that doesn't require, a laborious briefing book. But if we have those structural starting points already defined, and then we learn from the patients that we're treating, and we realize that we have to revise something around those structural elements, having that capacity and that mechanism to have that rapid feedback.
Travis, anything to add to what Rosanna said? And then I want to come to VJ and Stephen on that.
No, I think just going back to that framework and the ability to kind of have that framework come into a pre-IND meeting and kind of align on those things, whether it's specific to the trial, the DLT criteria, sentinel dosing, study pausing, but then having an increased level of confidence that when you submit the IND that those things aren't going to change. Because when those things change, especially for small biotechs and wanting to generate human data quickly and the funding resources that you have, having to go back through an IRB or back through contracting, it's almost like starting all over again. And so it's time consuming. But obviously things change between pre-IND and IND. The FDA, we provide a lot more information during an IND. But if there's certain things related to specifically to the protocol where you have an increased level of confidence that those don't change, you may have to answer more questions around the non-clinical package. And maybe that doesn't ultimately impact the protocol. But if you have more confidence going into the IND based on the pre-IND meeting that things aren't going to change, you could accelerate development in many ways.
VJ, Stephen, any reactions to some of these proposed solutions? What you think are important considerations?
Sure. So, as an office, we are open to exercise of any applicable regulatory flexibility as long as it does not compromise scientific rigor. So that goes without saying. And Nirali is absolutely correct that when you start the study, there are many unknowns and you learn about some of the things during the study, and we encourage sponsors to not only have early and frequent interactions, but to engage with us during all phases of the development. And one of the things that we routinely kind of see and is you are seeing some early efficacy signals, large treatment effects, and you want to kind of accelerate your clinical kind of development program. We continue to have challenges with alignment of CMC and clinical development timelines. In the oncology kind of lexicon, this seems to be a refractory relapsing issue despite multiple lines of prior communication. Okay? And I want to share a poignant anecdote, which kind of happens all too often. So we had a situation not too long ago where a company submitted a briefing package and their plans for a BLA, and our clinical review team and the CMC review team, based on their analysis and reading of the package, were about 18 months apart on when the company would come for a BLA. So we said, okay, there must be typos, there must be some things that's kind of going on. So we had a sponsor meeting. And what was surprising was these sponsors themselves were 18 months apart, and our reviewers, once again, were correct. And the sponsor tried to explain it away that, clinical study is handled in-house, but the CMC is outsourced. We cannot kind of have that situation. And there is a joke among some of the CMC reviewers that when an IND is allowed to proceed, clinical takes the bullet train and CMC takes the choo-choo train. That cannot kind of like, happen.
Okay. What do you see as potential solutions around for this issue? Is there a mechanism like Rosanna proposed of maybe more frequent interactions that could help the choo-choo train catch up to the bullet train?
Well, I think, we had the, phase 1, phase 2, phase 3 development, and now we are moving towards a default, single adaptive design kind of like, trial. So some of the core elements of the design should be alignment of the CMC timeline, and also, as I mentioned earlier, the timing of the pediatric enrollment. Those should be design questions, not like, I will wait for the phase 1 data, I'll wait for the EOP2, then I will think about a pediatric study, then I'll think about manufacturing scaling. So these have to be central design questions that the sponsors are aware, and take advantage of when they are thinking about a single adaptive study design and single pivotal study with confirmatory evidence.
Mm-hmm. Thank you for that. That's a nice transition into sort of our next topic that I want to cover, which is around the types of early data investments that have the greatest downstream leverage, and where gaps can create the most significant constraints in terms of timeline, efficiency, including on trial design choices such as single arm versus randomized trials, and use of novel endpoints like MRD and multiple myeloma. So Rosanna, from your perspective and a late stage perspective, which early data investments have had the greatest impact on kind of the registration stage options that you consider?
Yes, so our approach to early development design is to make sure that the drug is modulating the target and getting that initial proof of concept that informs late phase design. So it's not just exploratory, but with the single trial design concept, really looking at where we want to go, and so starting with the end in mind. I think what's helped us very much at BMS is really the investments that we've made in our integrative predictive sciences, machine learning, AI, and data analytics that were really enabled us to make high quality decisions at key inflection points and move things more efficiently along in the late phase space.
Travis, do you want to provide the biotech perspective on this?
I don't have too much to add. We don't have a lot of AI or machine learning and big data sets, but I think to build on what Vijay just said, it's really taking advantage of the FDA interactions, Type D meetings as early as possible. One of the biggest places you can't invest enough with cell therapies, knowing how quickly they can go from early phase 1, demonstrating incredible efficacy, to wanting to start registration trials and making sure the choo-choo train is actually on the bullet train schedule, right? And so you can't invest enough in CMC process development and engage with the agency in a meaningful way to be transparent about our plans for clinical and how we're going to advance from an initial process, or really can you make that initial process so it's really viable to go to those studies first, right? To go to those accelerated approval type studies without needing to do a process update. But even if you need to do a process update, when can you do it? Share that information with the agency, get feedback, and just have this informed both CMC and clinical pathway that's integrated, so you don't have these disconnects.
That's great. We're running short on time, so I want to quickly go back to you, Vijay, and ask you to comment on how FDA thinks about the relationship between, early mechanistic, biomarker data and downstream regulatory evidentiary expectations, and whether there are situations where the product type would influence the study design or endpoints.
Thank you. The core principle of evidentiary standards for biomarker obviously depends upon how it is used and the associated risks. Travis mentioned about accelerated approval. We have been using the biomarkers as surrogate endpoints for the accelerated approval. We encourage biomarker-driven enrichment for patient selection for if the early phase studies show promising biomarker data, target engagement, downstream pathway modulation, the expectation from us is that the phase 3 design is built on to explore to verify these kind of like, early signals. The other thing that I also want to say is that we support prospective biomarker qualification within the registrational study design. The ICH M10 is a starting framework for method validation and can be adapted for specific COU. In terms of the late phase kind of like, study, yes, we want to kind of like, have randomized control study, especially for those that are survival-based time to endpoints such as PFS and OS, but we know that those are not practical or kind of like, feasible for especially rare diseases and relapsed cancers, which are refractory to multiple lines of therapy. In the case of the hematologic malignancies, single-arm studies with a large treatment effect and a durability of response has been accepted for approvals with the CAR-T products. When it comes to solid tumors where the cell therapy is still to be established or less established, we require a more rigorous demonstration of clinical benefit, usually a progression-free survival or overall survival. And in that case, we rely on the early phase mechanistic data to show tumor penetration and persistence in the solid tumor microenvironment. Durability is another distinctive kind of like, challenge, especially when we compare allogeneic and, autologous kind of like, products. The other issue is that the post-approval long-term follow-up should be viewed as an extension of an evidentiary kind of like, framework. So when that is used, we can appropriately balance the pre and the post market data collection.
Okay.
Thank you.
So just to clarify, so you and others at FDA published a perspective in JAMA, I believe, recently on CAR T-cell therapy and FDA's expectations or CBER's expectations regarding trial designs and things. Can you give us a little, can you describe for us a little bit why you felt the need to publish that and whether that represents a departure from prior oncology, expectations or even precedent with respect to when a single-arm trial is necessary versus when a randomized trial is necessary?
Yeah, thank you very much. The purpose of that publication was kind of like, twofold. One of that is that we have approved several kind of like, CAR-T products to look back at our experience and share kind of like, our retrospective kind of like, view of it and how we viewed it. Mm-hmm. The second part of it was the same basis for the CMC flexibilities. We have been giving individualized kind of like, advice to the companies, and we wanted to kind of like, have a broader kind of like, awareness. Mm-hmm. So if there is a treating physician and they want to know, is CD19 better than CD20? Is allogeneic better than autologous or in vivo? So at some point of time, we would be kind of like, expecting RCTs to kind of like, address these types of kind of like, questions.
Right. I think the question is always RCTs conducted by whom, right? And, as you know, there's no comparative efficacy requirement that the FDA has. And in settings where there are large effects on response and there are durable effects on response, I guess the question is whether, because there's another CAR T-cell therapy approved in that particular setting, you would expect a direct comparison to that, or whether it's still appropriate. It may still be appropriate for many different reasons, which we may not be able to get into today, to still conduct a single-arm trial.
Yes, I mean, as I said, we are, when it comes to CAR-T, we are always open to single-arm studies with large magnitude of, treatment response and durability of the effect. Mm-hmm. But at the same time, if the patient and the treating physician are trying to make a decision on any number of kind of like, characteristics, whether it is the short-term safety of the products, the durability of the products, so it would be kind of like, valuable for sponsors to kind of like, look at those things and say whether an RCT would provide a better answer to the treating physicians and the patient so that they can make an informed choice on their treatment, because these toxicities are serious.
Yes, yes, absolutely. Or the NCI with appropriate, adequate funding could also run some of those trials. You would agree, probably, yes. So in the last 3 minutes that we have, I want to sort of give everyone a chance to sort of looking across all of what we've discussed today. What changes, whether it's trial design, whether it's the regulatory approach, early phase development structure, would have the greatest impact that we could accomplish in the near term, just from your perspective. 1 minute each person.
So I would like to first just let everybody in the audience that's watching it to submit their questions through the QR code that's available, or the link. But what a patient really wants is to hear from another patient directly. So, when you go to sign a consent and you ask, hey, can I please talk with another patient that has done this before, a direct peer connect, so they can kind of see straight from the horse's mouth, okay, what was it like, and answer all their questions. The rules, state that, no, I'm sorry, no, we can't. You could tell me all day long, what cytokine release is going to be like. But I had no clue until I actually went through it. But when you talk to somebody else that has done it, they can help, walk you through it. So a lot of the rules, when you're signing consents, again, back to regulatory, maybe need some more discussions to start having.
Sounds good. Nirali?
Yeah, 2 main points. One, I'm really optimistic about what's happening with cell and gene therapy in the pediatric space. If you haven't had a chance or didn't get to go to the FDA panel on the pediatric, it is all available. It was beautifully conducted, and I think it's telling you that, and as Vijay had said, it's really thinking about the disease context and the patient population. And as we develop new studies in pediatric patients, we should use that guidance and approach the FDA, and I'm really encouraged with all of that. And then the second point is, in terms of streamlining pathways, I think we need to have now foundational frameworks for DLTs, for other elements, what is necessary, because I think having that foundation will help with each trial development, but take away a lot of the extra time that's needed, and then you can personalize it based on that foundation.
And having that foundation available, easily available, not sort of tied to going and talking each time to.
No, easily available, because as much as I appreciate, come to us and all of that, which is wonderful, meetings take time to happen, and you want to come as prepared and as full a package that you can, so that you have minimal changes, and so that will help reduce time.
Yes. Vijay, we need more guidances, it sounds.
Yes.
Or even, publicly facing websites where you could sort of provide
Again, we can help. Like I said, we have a publication in AML. The community can help partner with developing these foundations.
Okay, great. Rosanna?
I'm optimistic, sitting here with industry partners, academia, and the FDA, that we could come up with a common framework with those structural starting points, and that would make a big difference. I think certainly within the trials that are conducted in the US. I know we haven't talked about it, but global convergence is a topic that we've talked about for many, many years. I know that it's limited by individual country regulations, and so there will need to be some flexibility or greater flexibility to get there.
Yeah, I won't repeat Nirali and Rosanna, but the common framework concepts. But again, the enhanced flexibility in phase 1 to be able to react to data, have a framework in the study for how you're going to do that with a safety review committee, and the flexibility on the FDA's part to let that happen, knowing that we'll have to come back and work with the agency on those changes and how to incorporate them, and looking at the data in aggregate. But that flexibility, I think, is really important.
Vijay?
Thank you. I would just like to make 3 quick points. We understand the challenges for product development, and there is a slew of modernization efforts that are going on in affecting every discipline. So we encourage all the sponsors to take full advantage of all of those things, whether it is Bayesian methodology, NAM, etc. The second point that I would like to make is that we have shown incredible flexibility, and we publish the CMC flexibilities. Our SBRA documents list all the regulatory flexibilities that was exercised for that BLA. And the last point is, again, I cannot emphasize enough for sponsors to come to us early and frequently for tailored feedback on all phases of development.
Thanks, Vijay. Stephen?
Yeah, so I'll keep this real brief. I know we've a lot of talking about clinical trial, but to take it back a little non-clinical, I think looking towards the future, just participation of more sponsors in academia in the development of NAMs is essential, including research as well as work to qualify or validate NAMs, could really have impact in terms of reducing non-clinical studies and getting more efficiencies in determining appropriate doses to take to trial and moving forward with trials. So I would just leave it with that I think there's a lot of advancement in the future and a lot of excitement about alternative methods.
Thank you. We have less than 5 minutes to take questions. I've been a poor steward of our time. Fail for me here as a moderator. But just the first question, I guess, for Chris and others. In the case of an otherwise incurable cancer, do you think we should allow patients to make risk-reward decisions to enroll in first-in-human trials, even when they are earlier in their treatment course? Is this something that should be addressable through informed consent?
Oh, 100%, absolutely. Like, statistics literally show that the faster you can get to a cell therapy, the better. My trial had numerous participants that were very heavily pretreated, but through accelerated approval, thank God, it's now available, and everybody that's able to get to it sooner than later, is showing better results. That being said, when you're signing a consent, clinical trials, especially a lot of phase 1, I know many patients that have had a lot of really good success. And if you're able to do a clinical trial first, and for some reason it doesn't work, then you have, all the other approved therapies and then, maybe other trials. But I would always encourage, go to a trial as soon as you can. I believe even the NCCN guidelines states that as well, that your success, trial is going to be a lot more than not.
Anyone have anything to add to that? 30 seconds.
Yep, I can talk about the informed consent process. I think it's really important for people to realize that it's a partnership between the provider and the patients and their families. We take a lot of time to make sure patients really understand what they're going through. To your point about patients, there can be accidental run-ins with other patients who've had it. There's patient networks. I think that the patient advocacy group is really important. But part of the conversation with informed consent what are the alternatives? What else is out there that's available to you? And in the case we were presenting, you have an otherwise incurable cancer, and so you can present all of the options, but I think we don't give our patients and families enough credit to say that they are capable. And so it's not us allowing them, but they are capable of making the decisions that are right for them.
Yes. And recognizing that regulatory is one aspect of the informed consent and the safety assessment, right? There's the regulators, but there's the investigators, there's the safety boards, there's the IRB that are all have a stake in making sure that participation in that trial is appropriate. So the next question is there a role for decentralized trials in early phase studies of cell therapies, particularly for academic studies? Any challenges and opportunities you can think of?
I would like to take the first crack at it. We are kind of in favor of decentralized trials for oncology and even for non-oncology rare disease space. There is a significant burden on the patients and their families for trial participation. So any aspect of the schedule of assessments that can be decentralized would be kind of very important for the patients. Of course, kind of like, there could be more data point collections with decentralized trials, and it may not be applicable in the oncology space. There is digital health technologies like wearables and other things that can provide additional kind of like, clinically meaningful quality of life kind of like, outcomes.
So yes, you would be open to that?
Yes.
All right. So the next question is, in recent FDA guidance on the use of MRD in multiple myeloma, CAR T was omitted. What would it take to extrapolate into cell and gene therapies, and how are sponsors currently using it? Feel free to phone a friend.
No. Yeah, some of the whole topic of measurable residual disease is a relatively kind of like, newer concept, and that's been kind of like, applied to a lot of different kind of like, product types and a lot of kind of like, different, malignancies. So we'll take that kind of like, feedback, and we'll go back and we'll look at it, and we'll see if we need to kind of like, update any guidances.
Yeah, I mean, I think that this particular guidance was developed based on tremendous work that was done as part of a multi-stakeholder effort to use MRD in regulatory decision-making. But that exception in the guidance was due to the absence of cell therapy studies in those meta-analyses. And so I think what sponsors want to understand is what amount of data would it take to remove that exception from the guidance, and how should that data be presented? Ideally, not sponsor by sponsor, but as a broader community. But we can come back to this as a follow-up point. Hopefully, FDA would be happy to participate in this. All right, great. So that was the last question. I want to wrap up and just say thank you so much. So sorry to have gone over time, but I hope that the panelists' potential solutions were insightful, and we welcome additional ones following the meeting. Thank you.
Thank you.
Thank you, Lola, and to the whole group for your very insightful comments as part of the panel. I just want to take a moment to add my thanks for all of you for joining us today. We have a great group of thought leaders that are participating in today's discussion, both in the room and several hundred that have already logged in online for today's discussion. So thank you for joining the webcast as well. I also want to add my thanks to the Parker Institute for Cancer Immunotherapy for helping to facilitate this series of discussions over the past several years. I hope it's evident the progress that is being made both scientifically as well as some of these logistical discussions that have taken place. And we've seen that over the past several months, there's been numerous examples of remarkable progress in the cell and gene therapy space that are reshaping oncology and other therapeutic areas. So with that in mind, we're grateful to have such a remarkable lineup of folks from across numerous different stakeholders, which will be critical in thinking through these issues throughout today's meeting. Before we move on to our next session, however, we're going to take a brief break. You can find coffee and refreshments out in the foyer where they were located earlier, and we will reconvene in a little less than 15 minutes for our next session. And given the large audience online, we will try to reconvene back here at 11:30. So thank you very much. Okay, if everyone could go ahead and start grabbing their seats, we'll go ahead and get started with our second session of the day this morning. The session coming up next is titled Operationalizing Risk-Based CMC Flexibility Across the Product Lifecycle. As I invite our panelists to go ahead and come up on stage, just a reminder that whether you're joining us virtually or in person today, you can participate in the discussion via the Slido app by using either the QR code included on the meeting material or provided in the chat for those of you that are online this morning. Members of our team here will be reviewing the submitted questions and monitoring them when you submit them throughout the discussion to be able to shuttle them up to our moderators throughout each of the sessions during the Q&A portion of the discussion. So now I have the distinct privilege of turning things over to Dr. John Conley, the Chief Scientific Officer of the Parker Institute for Cancer Immunotherapy, to moderate this next session for us. John?
Thanks very much, Jeff. This is it's a session that I think it really sits at exactly the right point during the day. You know, the sort of heart of the matter being a lot of the CMC issues that come up. And so I think that overall, when you look at the titled Operational Risk-Based CMC Flexibility Across Product Lifecycle, really what we want to cover today is how existing CMC flexibilities are operationalized from IND all the way through licensure. And we're incredibly lucky to have such an amazing panel here today with some just some phenomenal people. Starting with, to my left, Ben Benesky, who's the Senior Vice President, Chief Technical Officer at Allogene Therapeutics. Ben's got a career spanning more than 2 decades of biological manufacturing, where he's overseen product development, manufacturing across a number of different cell therapies, including at Allogene, where he's really moved from executive director to plant manager, played a pivotal role in design, construction of the entire pipeline, so it's wonderful. And then Mark Better as well, so principal consultant of FarmFlex, as well as core team leader of Cell One Partners. Mark is, again, a biotech professional with more than 30 years of experience in cell therapy and biologic CMC manufacturing. Most recently, he's advised companies through his roles in each of those different organizations. Prior to that, Mark has really led the process validation activities as well as analytical development and process characterization of Yescarta, which, as you guys know, first-in-class CD19 CAR T. Magdi's wonderful to see you again. SLab is a director of process development and really leads the effort of cell therapeutics and transplant laboratory at Mass General Hospital. His work really has focused completely on both manufacturing, the science of manufacturing, but also all the way into regulatory. So it's this line of sight from process development all the way to true clinical execution. It's a pleasure to have you here. Just as well as we've got from Iovance Biotherapeutics, clearly a leader in this cell therapy field. We've got a senior vice president and technical director in Arvind Natarajan. So Arvind's got 25 years of experience in the CMC field for cell, gene therapies, biologics, and vaccine, including bringing 4 novel products to market. So just spectacular track record. We've also got Alex Sassi here today. So Alex is the executive director of CMC and regulatory for AstraZeneca. So it's wonderful to have you. And so Alex brings, again, huge experience in the CMC field, over 25 years in biomedical manufacturing and focusing on both regulatory components as well as the CMC field. So it's great to have you. And then finally, we've got Kimberly Schultz, who is the director of Division 2 for the Office of Gene Therapy, so this is, and Office of Therapeutic Products at CBER from the FDA today. She oversees product review, pre-IND, IND, and BLA submissions for gene therapy products, so great coverage here today, and serves on the working group for advanced initiatives in CBER across the FDA. So thank you, Kim, for coming. So we've got a lot to cover today, so I just want to really kick things off. Again, we want to talk a little bit about this and get to the concepts of CMC flexibility and how it's operationalized. But let's start with that concept of what does CMC flexibility actually look like in practice across your programs, and where does the friction arise? Because there is friction when you take it into actual implementation. So I think we'll start with Mark, sort of give us that cross-program perspective and where does friction actually happen.
Yeah, thanks. It's a pleasure to be here today, share a few thoughts. I've been a firm believer for a very long time now that having basic process and product understanding is really the key to bringing products forward through clinical development. And in that context, one of the main areas where I see big conflicts is between the ability of staff in sponsor organizations to exert the time and effort to develop that process understanding, product understanding, versus corporate timelines to bring products to market. And in that context, one of the questions that I get asked regularly, I would say almost monthly, is in some form, what is it that I need to do to convince regulators that X is appropriate for implementation, whether it's process improvement, a raw material change, analytical method development, new specifications, whatever the issue is on CMC side. The sponsors often are confused about what they need to do, and sometimes it's a very quick answer, but more often I propose a response something like, well, what will it take you to believe that this change you want to implement doesn't adversely impact your product or potentially impact the efficiency of your product in a patient context? And that gets the sponsor thinking a little bit more clearly about what type of information they need to collect to be able to introduce things. So I think that this disconnect between wanting to move quickly and efficiently through CMC development, we already know our clinical colleagues are concerned that we lag behind. And at the end of the day, there's a lot of details in CMC development. So how do sponsors develop that information base, get that product and process understanding quickly enough, yet still be able to navigate the evolving changes that are required during the development of a cell therapy product. Regulatory flexibility is one component that helps, but really I think to some degree it's the mindset within the sponsor organizations about what the requirements are that's going to take them where they need to go, that it becomes a big challenge.
Sure. Arvind, this is something you're really familiar with, particularly in the autologous TIL space. Maybe you could talk a little bit about how flexibility impacts your process.
Sure. So at Iovance, we are working on T-cell therapy for the treatment of solid tumor cancers. We have the first approval for metastatic melanoma, and that's been available for a couple of years. And really, we're using sort of the same approach to try and use T-cell therapy to treat multiple solid tumor indications. And we're also working on next generation TIL therapies. And so the problem statement as we see it is, how can we advance the development of the newer products as well as application of sort of the same process to multiple indications based on the backbone and the learnings from the first approved application, right? So there are certain elements that you can certainly capitalize on and learn from that. Other elements that you're going to have to account for because of the change in the product or because of a change in indication. The friction ultimately comes back to, because it's autologous cell therapy, because you're looking at multiple indications, you're always going to have that variability in starting material as a key contributor. And the better we can try and understand that, the more information we can gather on a population level. Those are things that help us as we think about developing products for other indications and next generation products.
For sure. And just, Ben, how do you see this applying to your, working with allogeneic cells at scale?
Yeah, so at Allogene, we develop allogeneic CAR T-cell therapies for oncology and autoimmune indications. And one of the big advantages of allogeneic is scale. Mm-hmm. One batch can produce 100 or more doses. Our manufacturing facility has a capacity of potentially 30 to 60,000 or more doses a year at a low cost, $10,000 to $20,000 a dose potentially. But that advantage of scale, when we think about clinical development, means we're supporting an entire clinical trial with a relatively small number of batches, which means less data for setting specifications, or a lower end for development batches and data. So flexibility around specification setting and a life cycle approach to release criteria is a really important area of flexibility. Also, we're using gene editing components in ex vivo manufacturing, in a very well-controlled ex vivo manufacturing process. So the development and control of those gene editing components is an area of flexibility that's also quite valuable. And how can we take a risk-based approach to allow us to sort of rationalize how much work we do on the gene editing components to facilitate development there. And then the last area is continuous improvement. We're always looking to make changes to improve our products and our manufacturing platforms. So flexibility in how we manage that change and how we perform comparability exercises is a really valuable area of flexibility for us.
That's a big one. We should touch on that a little bit more. Magni, when you apply this to the clinic now, how does this flexibility actually translate?
So at the Cellular Immune Therapy Program, we focus mainly on developing home grown CAR T, I would say. So we take an approach from the discovery to the clinic, and then we apply those learnings back to our discovery pipeline at Harvard Medical School and Mass General Hospital. And in this process, we usually encounter two areas where we see some bottlenecks when we are transitioning from the animal studies to doing our process development, and then when we are doing our process development to GMP and IND enabling studies. And these create some friction points with with the development and where we would see areas for flexibility. As we all know that solid tumors are a big bottleneck right now, and pushing forward is what we are trying to do here. And to pinpoint, we work also with gene editing and trying to create a more tailored approach and personal approach in the area of CAR T by giving the patient not only like one single gene-edited product, but we are trying to push the limits and try to see if the patient could benefit from a pool of gene-edited products and that could be administered. And this is, of course, requires some conversation and some creation of areas of flexibility with the regulators on how we can define these products. Should we embrace the heterogeneity of the product and heterogeneity of the starting material that we're working with as well?
That would be an important advance. There's just no doubt about it. It's one that we've covered in these meetings before and hope to continue to push forward with. Just taking a step back now, just on some of the points that you guys were raising on specifications. So particularly cell therapy specifications are often set early with limited data that's out there, and yet they do anchor everything we talk about with comparability and quality throughout the entire life cycle. So how should we approach setting and evolving specifications as they change, as the program advances? This one's one I want to actually ask him to take on, particularly from a regulatory perspective.
Yeah, and I appreciate you giving us the opportunity to start this conversation from the FDA's perspective, because quite honestly, we were quite surprised to see the misalignment in understanding of how specifications should be set and maintained throughout a life cycle in the issue brief compared to that which we promote through our FDA review processes. And so in many guidances, including the CMC gene therapy guidance, the CAR T-cell guidance, more recently the cell and gene therapy Q&A guidance, and the CMC flexibility guidance, which we published yesterday for everybody, we do promote a life cycle approach to specification setting. And this is for different reasons for different products, right? If you have a product where you have a limited number of lots, it is difficult to set a meaningful lot release criteria for that. Or what I'm going to focus on is autologous products, where we agree you need to, during your clinical study, get experience across that potential patient variability in order to inform your commercial lot release criteria. Now there's always a nuance, right? So this is where the rubber hits the road. So I think that there's perhaps two places where this misalignment may lead to, where confusion and comments that are given may lead to this misalignment. So we do recommend that you set very wide acceptance criteria during your development. These are not restrictive criteria, but these are things where if your manufacturing process for some reason is outside of even a wide normal for that lot, come and check in. Make sure that the monitoring for the patient and the risk mitigation strategies are appropriate for that lot. The other place where it is that sometimes there's a disagreement on whether a critical quality attribute is actually a safety attribute, or if it is just one that's really important for product quality, right? And so I'm just gonna use an example from CAR T-cells, vector copy number. So as many of us in the room know, this is the average number of integration sites for perhaps a lentiviral vector into a T-cell across the population. Now we do not have a predefined limit on where your vector copy number can be, but as you can imagine, more integrations leads to higher clinical risk. And so what we expect is that information from your preclinical experience, from your engineering runs, informs what your initial lot release criteria are. But I want to be clear. If you have 2 engineering runs and both of them have about 3 vector copy numbers per transduced cells, we're not going to hold you to 3. We're also not going to let you move forward with 15. So there's a lot of room in between there, and that's where that conversation, I think, during the pre-IND meeting is informative.
Yeah, that's so important, and it's incredible to have that opportunity to continue to go back and forth with this conversation. Alex, just touching on this as well, how does this then impact things like life cycle planning in your experience?
No, thank you for the question, and thanks for the invitation for being here. And I'm glad that I'm speaking after Dr. Schultz, because she definitely set the stage in terms of how the setting of specifications is and what's expected from the agency, right? So what we do in terms of life cycle planning is really that we have the limitations on the method, right? We discussed already here. There are limitations in terms of understanding of the clinical relevance, understanding of your method capabilities. So when we set specifications early, there is going to be those broad ranges that you're going to see. So what we try to do, and I wanted to make sure that nobody's discouraged by those limitations that we have, right? We look into a tiered approach. So in your tier 1 is going to be really your non-negotiable parameters that you're looking at the patient safety. So you definitely have to demonstrate your sterility, endotoxicity. You're doing a test for replication competence vector. And then you go into your tier 2, where you're looking into showing your process consistency. And then in your tier 3, you're looking to attributes that are related to monitoring your process and ensuring that some of their parameters are reportable. Now, and Dr. Schultz may confirm that the agency, and not only the FDA, but other agencies, they don't like that you're going to have report results in your specification. Sometimes we have constraints and we have to put that, but it's always a preference that you're going to use a broader range or one-side limit. So that gives more assurance that you're following the process and you have more understanding. As you progress with your development, right, you're starting to have more understanding on your methods, you understand the clinical relevance, then you can revisit those specifications. So you can also establish a gated approach for reviewing specifications with your clinical milestones that allows for this cross-functional review board to really dive into the specs and see how you can improve your acceptance criteria and make changes that will be applicable. And of course, when you get into a post-approval phase that eventually we all wanted to be there, some of us are already there, we are looking into the real-world evidence that then be like a very important feedback loop to bring into updating specifications, and I'm sure it's going to be covered more in this discussion.
Yeah, that's great, absolutely. Ben, just getting to that real-world question, real-world constraints and with the limited data that's out there?
Yeah, and I think the guidance that came out yesterday and the life cycle approach to setting specifications and some flexibility with that is really helpful in real-world situations where in going into a first-in-human study, again, with maybe only 2 engineering runs or a very limited data set, setting a numerical specification on anything, you may end up, even if you try to set it wide, it may not reflect the full variability of the process. And I've certainly seen examples where we set a specification that we thought was appropriate, but then learned through further manufacturing that we had a batch that was outside or it wasn't outside, it was approaching that specification, and we thought, oh no, maybe there was a manufacturing problem or some issue, and it turns out it was just normal variability that, that specification is one we'll have to look at over time as we've gathered more data to change. So the flexibility to allow updating of those specifications. And the same thing applies for even commercial specifications. If we set specifications based on clinical experience, and that clinical experience was generated with a small number of batches, it's the same problem that we may find the variability of the process is actually larger than we thought. So having a life cycle approach to being able to consider adjusting or resetting those specifications as more data becomes available is a really important area of flexibility.
Yeah, absolutely. Mark, I wonder if you could touch on that as well, where this spec setting kind of breaks in practice and how we address that.
Yeah, Ben, Alex, and Kim presented a lot of information. I just want to make 2 quick points, though. One, if sponsors think prospectively about breakpoints where they need to have specifications reestablished or reset based on the accumulation of data, obviously you need to set some specs as wide as they need to be at early stage of IND. Prior to initiation of a pivotal trial, it's particularly important to reevaluate those specifications before you enter that pivotal stage, particularly for product-specific attributes like potency and identity. And then before you get into your process validation activities, you need to set acceptance criteria for that activity. It's a good time to look at specs. And then prior to commercial, obviously, you have to look at all of the information that's available at that time and come up with reasonable commercial specs. So those kind of stage gates might be important to think about prospectively. The second point I'd like to make is about, we've talked a lot about the product specifications for our cell therapy product, but there are a lot of ancillary materials that are considered drug substances that go into the manufacturing where the data may be really limited, like for viral vectors, gene editing tools, tools that are used like monoclonal antibodies to select or as part of a manufacturing process. And there we need to be really cautious of setting specs with limited amount of data. It's challenging.
Mm-hmm. Absolutely. Arvind, you've got great experience, looking across the sort of totality of manufacturing and clinical specifications. Maybe you could touch on that as well, just the same kind of question.
Sure. I'll echo a lot of the comments that I've already made in terms of the, starting with the broad specifications for clinical. Where we are at Iovance is, of course, in a post-commercial setting, and which is why the guideline document that came out yesterday is going to be, I believe, so impactful and so helpful, because it continues to promote the idea of life cycle management of specifications. It's not that once you have an approved product, you're locked in and there's nowhere to go. And certainly working in autologous cell therapy, we continue to gain experience as we treat patients in the real world. And having that specification reflect not only sort of the clinical data, but also the commercial experience, as well as the range of manufacturing experience, I think that's going to be very important in managing through in a life cycle manner.
Right, so incorporating that real world data back
Absolutely.
in, really taking a deep look. This is where we all want to get to.
And can I just make just an ending comment about post-approval changes to specifications, similar to how our clinical colleagues have talked about not leaving the pediatric space to your post-approval, kind of place to figure it out. With the FDA, we are constrained by needing to have your specifications, so your commercial product be representative of the clinical product in order to give patients a reasonable expectation of those label claims. And so I think that it would be important to have an ongoing conversation, both internally and then with the FDA, on what types of information you can collect during that clinical study in order to support the acceptance criteria that you think would be important for a commercial product.
This is a challenge even just for conventional development. But you were mentioning before the kind of pooled product life on. This sounds like an even greater challenge there, where you're in the more experimental setting.
Yeah, absolutely. I think this is a very challenging area for defining and pushing the limits when we are working with refractory diseases. And our experience usually when we are going to first-in-human trials are coming from healthy donors that we are working with and trying to develop these specifications early on. And in my opinion, I think the focus should be more on, are we collecting the right information in our first-in-human trial to inform our specifications for the next phases? And I think we put a lot of time and effort in defining the specifications. Well, I think we should put more time and effort in making sure that we have the right data collection and right testing that we are doing during the first-in-human, because this is our learning process when we start working with patient material, and especially when we are working on developing these kind of multiplex gene-edited product and more heterogeneous products. This is where I think developing specification early on can be very challenging, and I think there may be an area of discussion, an area of change where we can think of more broader data collection approach and looking specification toward maybe even during the clinical trial or toward the end of the trial.
Yeah, absolutely. This is, in this experimental setting, you really are pushing the boundaries on both sides, both on the regulatory side and then, of course, in the biology as well. Yeah. So just let's get down to the kind of nuts and bolts of in practice, and that is this idea of comparability. So what's expected, where's the friction? So manufacturing challenges, as you guys know better than I think anyone, are inevitable, but comparability requirements can create significant operational burdens, particularly at multiple stages. Particularly where situations are where conventional equivalency testing isn't feasible. We've heard some of that today. For example, allogeneic processes, situations in which we consume the entire cell manufacturing product, rare cells, for instance, in some of the rare cell therapies, as well as site transfer protocols in which the sending site no longer is operational. So all of these are big issues that are out there, and I'm sure you guys have encountered even more. And so the question is really, where does the current framework work, and where does it fall short with these kinds of situations? Maybe we'll just start with Mark.
Yeah, thanks. Yeah, comparability, this is of interest to just about everybody in our field. So the FDA published a comparability guidance several years ago, which is fairly clear on what a good strategy for demonstrating comparability could be. It doesn't seem like it's a requirement, but there's a preference to do split starting material comparability and use appropriate statistical tools to demonstrate that pre and post change materials are equivalence testing. And a lot of people, a lot of sponsors have taken that approach to demonstrate comparability when a process improvement is implemented, when a site change is undertaken. And I think it's fairly straightforward. But as you mentioned, there are examples where doing split starting material equivalence testing is not easy or impractical when there's a limitation on the starting material, which would make it not possible to do the split studies at full scale, or when that site is no longer, the first sending site is no longer available to manufacture material to compare to a new site. So in those instances, sponsors need to think of maybe more creative ways to demonstrate comparability so that you can fully understand that the material pre and post change is equivalent, but you can't do split starting material studies easily. So an expectation approach is where you have a group of batches that were made pre-change and a group of batches that were made post-change, and they can be evaluated statistically to see whether they have shared characteristics for what you believe to be the most important quality attributes. So that's one strategy, but by itself it may not be convincing. So sponsors may want to consider doing laboratory studies at one site at small scale using qualified scale down models, for example, if they're available, to look at manufacturing at a smaller scale so you don't need as much starting material, side by side with the appropriate analytical methods. So it's an equivalent strategy, but not at full scale. So laboratory studies that might be supportive that your expectation approach data are in fact realistic, and you could move forward with that. I think that this becomes particularly critical in certain applications, and I would think with the proper process and product information, the proper analytical tools, that you could look at a combination of developmental studies to support comparability in the absence of a clear equivalence test.
That makes a lot of sense. Ben, this is something that you've certainly faced. Maybe some comments on.
Yeah, I mean, building a little bit on what Mark said about both practicality and logistics. Especially for allogeneic therapies, performing a statistically powered paired donor study, you could be talking about having to do, half dozen or so pairs, so that's, 12 dozen or more batches, making many more doses than you would ever need clinically in your GMP facilities, for example, to do comparability for a change. So the cost of that exercise can become quite high and make it somewhat impractical for logistical reasons as well. Certainly the case where perhaps an initial manufacturing site is no longer available to perform those studies, but there's other logistical considerations. If you need to use fresh donor material, getting that fresh donor material to geographically dispersed sites can be challenging. Or if you think about what comparability might look like if you were in a distributed manufacturing situation where you wanted to enable many sites your sort of N of that paired donor comparability and to get statistical significance across a large number of sites could be quite challenging. So, that the approach has a lot of practical and logistical considerations depending on the specific manufacturing process. And I think some of the things Mark mentioned, alternative approaches to use other process development data or other statistical or equivalence approaches could be really helpful.
Arvind, just picking up on what Ben just said about statistical variability, how are you approaching this distinguishing between patient variability and process analytical variability?
And that is the key question, particularly as it relates to autologous cell therapies, where you have limited amount of starting material. Some of the challenges that were mentioned in terms of if you start with fresh material or if you have sites that are geographically separate, how do you go about doing a split donor model, right? And so we have to think through that. So from a statistical standpoint, the key issues that we see are ultimately how do you decide what is a good set of acceptance criteria to determine comparability. And the choice of those acceptance criteria goes back to two key elements. One is what are you choosing as your reference data set, which may not necessarily be sort of your, let's say, your sending site. It could be an alternate data set, but what is your appropriate reference data set? And then the second question is how do you distinguish process variability from starting material variability? You can usually get a pretty good sense for the method variability from the method validation, but particularly for autologous products, how do you distinguish between the starting material and the process? And that then gives rise to some of the approaches that were mentioned, doing small scale studies where you can, and developing a scaled down model and then establishing some understanding of where the variability lies and then extrapolating it to a full scale situation.
Absolutely. Mark did a great job highlighting many of those. Kim, what's the FDA's take on this and the approach?
Yeah, so I don't think that you'll be surprised that I'm a huge fan of split-run comparability studies, and I'm not just a fan of them just for comparability, but I actually think in the data that we've received across a number of different files, that the amount of information that you can get on your process, the control, the robustness, the relationship of different parameters throughout manufacturing to endpoint CQAs is a wealth of information. So I actually recommend that when you do use split-run studies, that you use them for as much information as possible. And I have actually started to see people incorporating split runs into their normal development processes. So just a plug for that, even though I understand that there's logistical issues. And so as you would imagine, we have run into those a number of different times. And so it's difficult to give specific advice or general advice, I guess, without knowing the specifics of the situation that you're in. However, things like small scale studies in order to understand the actual impact of the change, when the change is occurring in your development process. If it's early on in development, our expectations for comparability are much lower than if it's happening after your pivotal study is done and you're moving into commercial and you're going to have no manufacturing experience at this new site. And so all of those types of things go into our decision. And then lastly, the amount of process characterization that you have in order to appropriately rate the risk of those changes on the actual manufacturing process and the resultant product is extremely important for our understanding of different strategies that you might be able to use in order to implement those changes.
Kim, just picking up on that and taking it forward, maybe is this concept of prior knowledge, and bring in its application to manufacturing. So where does the use of prior knowledge or platform data meaningfully reduce the burden in CMC?
Yeah, so prior knowledge, it's a term that's used broadly, but then there's very specific places where it can be used, whether it's your specific knowledge of a manufacturing process that's maybe used to make variations of a similar product. So going back to CAR T cells that are manufactured using the same process but target different antigens. And so then you would have a wealth of knowledge, I hope, with your process characterization and being able to leverage that. The other thing that we see is partnerships between different developers and being able to use that information in order to be able to inform their risk analysis.
That makes a huge amount of sense. Alex, so how does that platform approach, how is that applied now to large pharma CMC?
Yeah, so definitely we do look a lot in terms of the platform approach and prior knowledge, not so much in the platform designation as per the FDA definition, but our internal use of that knowledge, right? And there are instances where it's going to significantly reduce the CMC burden. So you can look into, if you can think about 3 examples. One is the manufacturing unit of operations, where you use the platform knowledge, you use the prior knowledge, and in a way that you can have an understanding of what's going to be your operating range parameters, and you are able to start your process already with that knowledge in place, right? The second one would be with your analytical methods. So you already know in advance if the method, what's going to be the performance characteristics for that method for when you're looking to the same modality. And the third one would be on your control strategy, right? And if I dare to say, you can even use AI in terms of establishing templates and establishing ways to look at your critical quality attributes in a way that you have that knowledge and you use that knowledge. Now, there are still limitations, right? Even within the same modality, biology sometimes wins. And you are looking at a different target. You have the variability of the cells that are coming from the donors or the autologous with coming from the patients. So you are aiming to use that prior knowledge, but then you get surprised. So you have to still look into what's going to be specific for that product. So the advantage is it can get a good starting point.
Yeah.
It can help you to minimize the bridging studies that you have to do specific to that product. And then you can also focus your efforts on what's going to be specific for your new program.
Sure. And this is also true for allogeneic therapy. How are you guys seeing this?
Yeah, I maybe 3 examples of where leveraging prior knowledge kind of helps get over that small data set I was talking about, and where the flexibility around that's really valuable. One, are evolving our manufacturing platforms. So as an example, we had some changes that we implemented to improve our formulation and filling processes for one product, but then implementing that same change on subsequent products that follow that same manufacturing platform can leverage some of the development data and experience we gained on doing that with the first product. Another area is in stability, and again, for products that are highly similar, manufactured on a similar platform, looking to see how we can use data to perhaps reduce the number of stability time points or the tests that are performed at each time point for subsequent products. And then a third area is maybe looking at product quality attributes that are common and maybe not product specific, and being able to use data from multiple programs to help generate a larger data set to set a better specification. So I think those are some examples of where we see advantages of being able to leverage data across platforms.
That's great. This is an important area, and so I just wanted to continue on this a little bit. So Arvind, how does prior knowledge of you can be leveraged for autologous on the same platform now?
Yeah. So I want to recognize a couple of points that Alexandra made. I mean, the importance of having a risk framework sort of underlying how we think about operationalizing a platform mindset internally, I think is very critical. The points that Ben raised about starting with the even if you demonstrate that the sample matrix is suitable for various analytical methods across different products, that itself gives you an indication that that fundamentally the underlying biology looks the same. I think it's important to look at the signals that may come out to say, if it doesn't quite look the same, maybe there's something else going on and you need to do a little bit deeper dive. But if that checkbox gets checked off right up front to say, yep, it looks the same, a T cell is a T cell that behaves the same under a certain set of conditions, then you're sort of halfway there. But to use that risk framework to really think through that process and to establish that in a very methodical manner, I think is key to being able to leverage all of the prior knowledge.
Sure. And Magdi, just finishing off here, so from the sort of from a more manufacturing science perspective, what is the critical data that's really required to get a meaningful extrapolation across different platforms or cell therapy products?
Yeah, for us, I think it was, we as an academic institute, we are lucky to have some prior knowledge that we built over the years, opening like multiple clinical trials. And this helped us like develop some knowledge that we have been using now to advance some of our programs forward. But I think it is important from the regulatory side that we, as academic centers, get more tailored advice when we are reaching for pre-IND and IND meetings, understanding that some academic institutes did not have like the regulatory experience building their program yet, and they did not accumulate the prior knowledge that we are talking about here. I'm happy that I'm sitting within like industry groups that have the resources to build these kind of data sets and build this kind of knowledge. But as academic institute, we might need some tailored advice based on the FDA prior knowledge that they can help us establish these kind of metrics and push our development forward.
That makes sense. Just one consistent theme we see across CMC discussions is whether current expectations are appropriately calibrated, to the actual product and patient risk. So just a quick question here, where is the current framework well calibrated, and where does it impose a burden without commensurate benefit? Maybe start with Mark.
Yeah, let me address that, thinking about process validation. So the historic strategy for process validation is to do multiple lots at scale, collect data, move forward. And I think for cell and gene therapy, particularly cell therapy applications, there are still examples where that kind of a strategy is appropriate. For example, if a sponsor is manufacturing viral vector for use in manufacturing a cell therapy product, or maybe an allogeneic example, it may be useful for companies to do 3, 5, even more lots as part of their process validation PPQ campaign to demonstrate that their process is consistent, because you can take that material that you generate and put it into inventory for either clinical use or commercial use later after approval. And so that makes a lot of sense. Then I think where people need to be a little bit more creative and look for some flexibility on how they do that, some examples might be, or it might be things like if you're making guide RNAs for use in gene editing, or an enzyme that's going to be used for part of your manufacturing process, Cas9, for example, or something similar, or other types of products like monoclonal antibodies that are used for cell selection. The cost of a manufacturing lot might be in the millions of dollars. You may not need the material for multiple lots to support your manufacturing opportunity. If you make multiple lots, some of that material might expire before it could be used. Mm-hmm. So it becomes a little bit impractical to consider making many large-scale lots as part of a process validation campaign for a cell therapy product. But there may be alternative opportunities there that are a little more creative, that give you the same kind of information base but are a little bit less egregious. For example, can you have a qualified scale-down manufacturing opportunity for a guide RNA or a viral vector, and you support process validation with, say, one large-scale lot and several, I don't know the number, but several small-scale lots that would not put you in a situation where you had excess inventory, wouldn't necessarily be prohibitive from a cost perspective. And I think that if you have the right kind of process characterization data to support your campaign is appropriate, if you have the right analytical methods to support that you can effectively identify the quality attributes accurately, and you have scaled-down models that you believe are representative of a full-scale lot, maybe a creative approach like that would be appropriate.
Absolutely. Magdi, where do these expectations maybe diverge in practice?
Yeah, I mean, I think we logged the process in on certain things like early on. I mean, to the point when we are working as an academic institute early on, we working on developing, for example, gene editing techniques, and we moved from the preclinical and we are starting to do our GMP manufacturing, and we realized some of the reagents that we have been using, there are no GMP equivalent, and and now we need to, for example, transfer to a GMP equivalent reagent, and then we get into the idea that we need to generate these, for example, Cas9 RNA that we need to manufacture a lot for a very big, large amount of money, and this creates some prohibitive environment for some of the innovations that we are trying to do, and this is where I think there is area of understand or like discussions where, for first-in-human academic sponsors, where we can set the limits for some of the intermediate reagents that we are using. For example, guide RNA, can we produce like batches and qualify them in a certain way, and working with the regulators on some of these specifications, and also like, for example, for some of the new gene editing methods that are being used, such as base editors, where the big manufacturers did not come in yet and make their GMP reagents for it. Can we actually generate this in-house and work with the regulators on developing some minimum specifications that we could use at this phase-appropriate stage to move things forward?
Again, the importance of communication. Kim, how does the FDA approach this sort of risk assessment?
Yeah, so as you know, we're a science-based organization, and so therefore we're going to assess the risk based on the information that's provided. And so PPQ runs, if we're talking about those in particular, that's one part of process validation. And so really, I think that a lot more emphasis should be put onto process characterization and understanding your process, and then using that information in order to justify your PPQ campaigns. Now I get it. Like, you're a small company, you're a startup biotech, that's expensive, and that's time consuming. So I'm just gonna put out a creative thought that we have seen be successful where multiple sponsors have worked together. They are using a similar process or the same critical components across products, and they've formed partnerships in order to have that process characterization, have that pre-work done that's necessary for it, even to be able to share data across. And so in order to leverage, they have to bring this information into each of their INDs or cross-reference it between the different ones. And it's been successful both in IND stage and then moving into later stages as well, this information sharing between different sponsors to have the necessary data, but to kind of split the cost and burden.
That's fantastic. Yeah, I'd love to see some examples of that out there. That's wonderful. I just want to finish up with one important question that I want to go down the line, which is that we did cover a lot of things today with specification development, comparability, platform frameworks. So one question I have is really for everyone now. What single change, whether it be in guidance, regulatory approach, industry practices, or academia for that matter, would have the greatest practical impact on CMC predictability for cell-based therapies? And Ben, I'm throwing that one right to you first.
Thanks.
You bet.
So we've talked about a lot of things that are already in some of the flexibility guidance that are really valuable. I think one more thing, or one thing that we could maybe continue to think about and add to, is risk-based approaches for the gene editing components that are used in ex vivo manufacturing. You know, you're seeing more and more therapies with multiple edits done. And if each one of those gene editing materials, if there's a way to be more risk-based and think about how can we reduce the requirements, it might facilitate developments of these therapies. You know, even adding one more test for one of these components, developing that method, qualifying it can be hundreds of thousands of dollars and months of work. And I worry that that may discourage some sponsors from moving forward with these multiplex edited approaches because the requirements become kind of burdensome. So I think continuing to look into risk-based approaches that don't sacrifice safety, that they're still scientifically rigorous, but could reduce those requirements is good. And since I'm going first, I'm going to be greedy and pick a second thing, which is continuing to look at harmonizing requirements across regions. There's many areas where FDA is quite flexible in how we approach these things and where other regulators are less flexible.
Sure.
And so you kind of have to then conform to the highest bar sometimes. So I think the more that we could harmonize these flexibilities we see in the US with other areas, that could also be helpful.
Mark?
I think we need public meetings where sponsors can get aligned on things a little better. Maybe it's happening, but I don't think it's happening fast enough. I think there's a lot of lack of clarity within sponsor organizations about when comparability exercises are required versus just old-fashioned process development, characterization data to support IND amendments.
That's great. Thank you. Maggie?
I mean, I think Ben nailed down what I wanted to say as well. Exactly. It's just like, and take this and look at it, and then look at our scale. Sure. when we are working with federal funding and trying to push the boundaries forward and then we are met by some expectations that at our stage and at our scale is sometimes challenging. And I think this is the biggest area. I think we came a long way in terms of standard CAR, I would say standard or traditional CAR T development, but now we are opening a new frontier where we know that CAR T might require more than just a working binder, and it might require not a single gene edit, it might require a pool of edits or multiplex gene editing. And then when we are thinking about this approach and trying to anchor the specifications and the characterization of these products to the same standards that we developed for a traditional CAR T product, this is where I think the biggest area where we want to sit down and discuss how we can push forward, maintain patient safety, which is the biggest concern in this stage, but also offer some new developments and push the boundaries a little bit.
Thank you. Arvind, what are your thoughts on a high-impact single change?
High-impact single change is continuing the, from the flexibility document, the lifecycle approach to specifications, extending into a post-approval environment. We're all aware that specifications tend to tighten over the course of development. And in a commercial product, if you have OOS products, there are avenues for patients to get access to those products, but it's typically, you have multiple sources of friction there, which ultimately limits the availability of those products. So having this life cycle mindset for specifications, I think is going to be hugely impactful.
You have an important perspective here, so
yes.
I'd love to.
So I would say platform frameworks that can be transferable across programs within the same modality and could be a starting point for companies to get ahead in terms of that prior knowledge. And in addition to have that as not only within the FDA, but harmonized under ICH, so for global submissions.
Okay.
Yeah, and so I think from our perspective, we heard a lot about this with leveraging and the use of prior knowledge. However, we're constrained in being able to share proprietary information. You don't want us to go out and tell all your secrets to everybody else, right? However, we take that information and we try to provide guidance in venues like this on ways that can be done. But I would really recommend that for the field as a whole to move forward, that perhaps we look into non-conventional partnerships that can move at least these early stage issues forward or come up with ways to work around some of the issues that we see in the field. And so for different reasons perhaps, but maybe look into the frameworks that were set forward by either the Bespoke Gene Therapy Consortium or the Somatic Cell Genome Editing Consortium. They're really trying to work as a field in order to solve these problems.
I love that. This kind of public-private partnerships would be really important. Well, I think we've got a little time for some questions. Once this we're deep into conversation, I wasn't even looking at the time, so terrible. But the first question is, could there be an opportunity at the time of approval to prospectively define acceptable ranges or parameters for certain specification changes so that programs can evolve more efficiently in the post-market with agreed guardrails without requiring repeated renegotiation. And that's for everyone, but Kim, I'm going to throw that to you.
I was going to say, I'll start it off. We do try to look at different approaches that are available depending on the type of product that you're looking at. And so I think one of the things that you'll see in the CMC flexibility is that when there is a limited amount of information at the time of approval, and this would be particularly pertinent to critical components that are used in ex vivo modified cells or direct in vivo vectors where you may only have a few lots at the time of approval. And so we exercise flexibility where available on those specifications, the acceptance criteria. And then you can see through approval letters that are public that we then have commitments in order to reassess those when there is additional manufacturing experience in order to make sure that the experience that was gained with that limited number of lots is appropriate for a larger experience with that manufacturing.
That makes sense. Second question, can anyone share experience as FDA or sponsors with the Platform Technology Designation Program? Are there opportunities to better operationalize this program and apply it more broadly with or without platform designation? Anyone want to comment on the program or their use of it? This is just Kim and I chatting.
Okay, I'll answer again. And so the platform technology designation is really a formalization of leveraging data, right? And so this kind of changes the risk for that particular designation where it's already been reviewed, it's already been validated, and it's in a licensed situation. But I think that the idea of operationalizing, leveraging is really what we already do, and we've talked about a number of times, and that's being able to use information across different files. And so we see this with cross-referencing with different files or with sharing between different companies. The difference is that we haven't necessarily reviewed it or we haven't agreed to that it is applicable to your product. So you need to provide that information in a description of how it's being used, and then we'll look at it and see if it is applicable.
That makes sense.
Yeah, maybe I can add in too. I think that sort of informal sharing of information across products and platforms, which is certainly easier when it's all within one company and you don't need partnerships to do it, is something that's already acceptable, based on the scientific data you can generate across programs. And when it comes to the formal platform designation, we haven't gone that direction, but have discussed, when would that be appropriate? Do you do that for the second product, or at what point is that platform designation going to be valuable to a company? I think where we've seen it be more used are for maybe I don't know if service provider is the right word, but maybe not by the therapeutics company, but by somebody who has a technology platform that they're looking to leverage across multiple companies or multiple sponsors. So I think thinking about when is that formal designation going to be beneficial versus continuing to maybe more informally leverage data across your platforms.
That makes sense, Ben. Thanks. I think we'll have one final question here is, how must CMC frameworks evolve as we shift from cell as a drug to body as a factory? Specifically, how do we define control and release a product when manufacturing is no longer confined to a facility but occurs dynamically within the patient? This is clearly an in vivo kind of CAR T question. Anyone want to pick that up? I'm going to pick on you, Kim. Any thoughts on in vivo CAR T and how this then applies to this concept?
Yeah, so as the space is evolving, we do see this question quite a bit. Oh, it's being made in the body.
Right.
But this is really no different from a regulatory standpoint in any other vector gene therapy. And so the drug product is the product that's being delivered to the patient. And then the data that you accumulate during that clinical study is what is going to provide the data to show, to support the clinical efficacy and safety of that product for licensure for your intended patient population.
Fantastic. Well, I want to just finish now by thanking the panelists for the really enlightening discussion. It was fantastic, and thanking my partners at Friends of Cancer Research for putting this on. So thank you.
Thank you.
Thank you.
Great. Thank you, John, and thanks to all of you for a terrific discussion. In the spirit of efficiency, we're going to try and operationalize that again here at lunch and take a 30-minute break. and hope that everyone online, you find time to do the same. For those of you that are in the room, there should be 2 stations for lunch outside of the doors back in the foyer. We would welcome you to bring your lunch back here, take a few moments to network with your colleagues. But please attempt to be back here at 1 o'clock. We have a really great lunch program with a couple of different leaders that will shift into more of a policy discussion more broadly about the future. And we hope that you will join us in a half hour. So thank you all.
Getting close to 0.
Okay, I think while people are finishing up grabbing lunch, hopefully, please feel free to continue eating during our keynote session here. But as we transition to our fireside chat this afternoon before our final session, our goal here is really to explore some of the broader policy dynamics that are shaping drug development and investment more generally. So we may veer a little bit from the cell therapy theme today, though by all means, we have 3 wonderful panelists with us today, so we'll let them guide according to their expertise and insights. But I'm really pleased to welcome to the event today Chris Heery from Acellus, David Kaufman from Third Rock Ventures, and Julie Tierney from Levitt Partners to help us dive into some of the details today. I won't spend too much time going into their backgrounds. That's all available on your meeting material should you want additional information on their bios. But suffice to say, we have a great group of experts to help us dive into some of these issues. And given our time, maybe we'll just go ahead and dive right in. So, thinking about some of the themes that have been presented in greater detail this morning, I think we've seen both in the cell therapy space and in the broader therapeutic landscape more generally, is that there is a great deal of momentum in oncology and in other therapeutic areas, that the science is really accelerating. But it is in the context of an overall environment that is facing a level of policy uncertainty that perhaps we haven't necessarily seen, at least in recent years. Things like NIH funding pressures, the evolving drug pricing dynamics, FDA leadership and staffing changes, and even growing competition from other parts of the globe are all sort of having a confluence factor simultaneously here. So I'd love to get your thoughts on how you're thinking about that particular combination of events and how companies, investors, and institutions are navigating these areas. So maybe, David, I'll start with you. So given sort of the uncertainty that is being encountered here, if you are a watcher of the trade press, it isn't uncommon to see things like acquisitions occurring at a pretty remarkable dollar amount. So I'm curious, what signals are investors watching? What's providing confidence or hesitation as you think about sort of a long-term approach here?
Yeah, so Jeff, like you say, when we're thinking about building a company, ultimately what we're trying to do is to find really important innovations that can meet big unmet medical needs and get value recognition for them. And that either happens, as you're pointing out, through strategic M&A or through public market recognition. And at least right now, as we're talking about, we're in an era where there's been a lot of good M&A activity recently. There's a huge amount of pressure from patent cliffs for this to occur. And so for the foreseeable future, it feels like we're in a healthy market for that kind of value recognition. We could talk for a long time about public markets, but let's say that they're slightly healthier than they were recently. So those factors are there. But some of the uncertainty of the environment on different time frames are going to intersect to create more uncertainty, right? So we have to start with great ideas. Great ideas come globally, but also very much from relationships that we have with investigators. And so the less healthy our academic innovation ecosystem is, the less we are being efficient at sourcing innovations that could lead to breakthrough medicines. We're also in an era, I think, and we can talk more about this as we talk about China later on, where first mover innovation is still getting rewarded with pretty high values. But what you see is a huge wave of competition from China coming very, very quickly in every modality, including complex ones, right behind those first innovators. And so the value of a second or third mover is much less as a pharma is thinking, well, do I pay a lot for that second mover, or do I wait 6 to 12 months and just find something much cheaper in China? How those dynamics are going to equilibrate, because we're in a period of disequilibrium around all that, is still quite unclear. And then, on top of that, we, in addition to thinking about value, we think about time and cost to get to value. And that's where the whole regulatory innovation space comes in. This is an ecosystem that I think the faster it moves and the more certainty it can provide, the more companies can understand their cost-time value equation.
Chris, maybe a similar question from your vantage point. How do clinical stage companies navigate or modify their development strategies with multiple different variables sort of at a macro level shifting at once?
I think it really depends on what the therapeutic area you're working in is. So if you're in cell therapy today as an early stage company, you have to have a product laid out to its commercial use case to attract investment dollars. Investors don't want to be on the ride of finding out if a platform works and then figuring out what the indication is going to be and then trying to figure out what the commercial output is going to be. They want to know what that plan is to start from. So I think part of the reason we're seeing fewer and fewer autologous cell therapy companies starting out right now is that you're seeing a consolidation in the cell therapy space for autologous, because that total quantity of created batches actually is what creates the leverage to be able to make new therapies. Because once you make a new therapy, if you're a baby biotech company, you still have to raise $1.5 billion to think about how you're going to commercialize that. And the dilution that you're going to cause to your investors is just too much for most investors to be interested in, which is why I think you're seeing investment dollars a bit more interested in things that are off the shelf that can be made in high quantities.
Julie, you've had the vantage point from several different angles, particularly in the policy space, on the Hill, at the agency, now as an external observer of all of those things. So when you think about this current policy environment and beyond FDA shaping, how are some of these beyond FDA issues shaping a current environment for innovation, and where are you seeing Capitol Hill take notice?
Yeah, I mean, I think that the Hill has been busy with a lot of macro issues and sort of bigger healthcare issues, but I think there's a number of factors sort of coming to bear that are ending up with more interest in the space. I think with all of the changes at FDA and NIH and sort of within the federal biomedical ecosystem, there is sort of attention because of that chaos. So we saw the Aging Committee in February do a rare disease hearing and try to sort of extend their jurisdiction over some of these FDA-related issues, and some specific members taking interest. Representative Auchincloss from Massachusetts, Senator Johnson as well, sort of wanting to dig into what is being presented to them as changing advice at FDA and lack of access. And then I think, as we head into the user fee reauthorizations next year, which will come up on a deadline to be reauthorized in September of next year, FDA and industry have just about finished with their commitment letter negotiations, and then that will sort of springboard and be transmitted over to Congress with the opportunity for policy riders. And so for the committees of jurisdiction, energy and commerce, and the HELP committee, they're starting to gear up as well to think about what are the policy riders that they may want to attach that are going to address some of these issues, perhaps provide stronger guardrails around processes at FDA, give some direction to FDA as they move into emerging technologies. I think keep a close eye on that, because that's going to start to accelerate as we head into next year. And then, of course, there's likely to be a shifting oversight environment, and we see folks interested in that space as well.
Yeah, maybe just to follow up on that for a second, given your past roles in prior user fee reauthorizations, I think the negotiation timetable has stayed relatively on where one would expect them to. How challenging do you think this will be on Capitol Hill, both in terms of macro issues, but even in a midterm year?
Yeah, so I think that traditionally FDI issues have had a lot of bipartisan support. I think there's more partisanship than we've seen in past years, but this is one of the few pieces of must-pass legislation, so I do see folks coming together and well, I don't think it's going to be a clean user fee reauthorization. Sometimes people throw that out, and that would be sort of like get it through, and that would be great. I expect it to be somewhere in the middle. Some important policy writers definitely included, and I think that I'm hopeful that given the fact that the negotiations have gone relatively smoothly, at least so far for the prescription drug user fees, that there will be congressional engagement to move that forward.
So let's shift back to some of the points that were made around globalization and advancements in innovation in China that seem to be getting a lot of attention, rightfully so. And you mentioned, David, that while this has often taken the shape of sort of follow-on type development or next in class, in some ways that's starting to begin to shift, where there is increased success in first in class development from other parts of the world. We've also seen that this has led to a continued increasing environment in terms of investment in a more conducive environment than perhaps previously seen in China. So I'm curious how you all are thinking about these particular changes. When you think about where innovation is happening, who funds it, and whether it is a threat, an opportunity, a mixture of both. So maybe, Chris, I'll start with you. How do you see this? As a threat, opportunity, both?
Both, for sure. We were talking about this just before we came up. I think if you're looking at a truly novel technology, something that you believe works in a certain way because you've identified a mechanism of action in the preclinical setting and you think that that should work in a certain way in a human being, I think it is a big leap to go to a geography where the controls are fewer. I actually applaud that FDA has a lot of rigor in what is granted for an IND. Now, we could talk differently about on the back end of the approval process and maybe some of the restrictions on how labels are given, et cetera. I think that's a different conversation, but having rigor about what is allowed to go into a human being when there is an uncertain safety impact and no known efficacy, I think is appropriate. So in the meantime, if you're in a situation where there's no other way to evaluate that question and you have to put it in a human being, I think that is where you're seeing a lot of companies use the opportunities in China and other geographies where you can try that with less restriction. But is that a threat? Is that an opportunity? Yes, it's both. But I will also mention that some of those things that are threats, we don't even fully realize until sometimes those drugs have actually been brought into the United States, have been evaluated, not just the United States, but Europe, et cetera, where restrictions are a little greater. And then we see that there are toxicities that were not fully characterized. So I think those are things we really need to keep an eye on. I know we get excited when we see data that looks impressive, but we also need to keep the caveat that sometimes there's other things that we're not seeing.
Yeah. In terms of sort of this shifting approach, have you seen this be a very rapid reaction in the biotech industry or something that's just sort of evolved over time in terms of doing studies, some initial studies elsewhere?
Well, I mean, now we have multiple agents that are approved in the United States that were first developed in China. So I think it feels like it's all of a sudden, but I think those of us that have been working in the space know that this has been coming for some time. So I think there's a more rapid pivot now to try to use that opportunity for certain companies in certain situations than there was 10 years ago. But certainly, I think that was already happening 10 years ago, and certainly 5 years ago, we saw plenty.
Yeah. So is it fair to say with sort of the time and cost savings that doing business to some extent in China is a necessity?
So maybe a better question for David.
Yeah. I mean, maybe to start with, I think any answer to these questions today is going to change almost at the speed of light. And I think for a policy forum, it's important to understand the real critical challenge of how does policy keep up with the just incredibly rapid changes in this ecosystem. I mean, even to the question of like the quality of clinical data and such. I mean, pharma are voting with their feet right now. 35 to 40% of new drugs in the pharma pipeline are in-licensed from China, mostly on the basis of clinical data. And so companies are trusting the quality of the clinical data enough to make pretty major strategic acquisitions. We're also in an era where, if we do get, say, PD-1 VEGF bispecifics approved here, the US won't have had the first shot at that, right? We'll be following China in getting those drugs to patients with a several-year delay. So that's a really, really different era that we're in right now. I'd like to go back, though, and say, I think that there is a huge amount of opportunity to win for patients if we don't really think of this as sort of a zero-sum game. When you have a lower cost structure, and when you're able to get to patients more efficiently, and you have just a huge volume of patients, you can ask questions in the clinic, hopefully done under appropriate, ethical standards and controls, that otherwise might not be able to be asked, right? So let's say you have a new autoimmune drug. It works by a mechanism that could be relevant across tons of autoimmune diseases, but you don't know which one it's going to work best in. You don't know if there are biomarkers you need. You're not sure about combinations. That kind of clinical iteration is something you can now do with the China cost structure. And we just, last week, one of our portfolio companies, Candid Therapeutics, was acquired on the basis of, this is a company that's making T-cell engagers for autoimmunity. Lots of people are doing that. But what this company did was establish just a clinical wall of data in multiple indications. A large portion of that was done in China, not exclusively, but I think they had 9 studies open across different indications in China, all enrolling. And that's kind of the basis for the acquisition, was just being able to get ahead of the pack with that level of data across so many different indications. And so I think, patients win if the data end up being, reproducible in global studies, and with all the right caveats that I think that you're bringing up, Chris.
And Julian, thinking about some of the different policy proposals that have been floating, to the point of a very rapidly changing environment, yet an increased level of attention from various congressional offices in thinking about how to appropriately address this particular environment. Things like that have been proposed around expansion to the Biosecure Act as one potential example. I think there has been and will be some debate around appropriations language, looking at the potential utilization of Chinese data in review or licensing applications to the FDA. So how do you see Capitol Hill reacting to this? Is it going to be rapid? Is there thoughtful discussion?
Well, I think sort of the bingo card. Do you put China and competitiveness on? Maybe we could throw in AI and we hit those. Bingo. But yeah, I think there's a lot of attention to it, and a lot of attention at FDA with, we've seen pilot programs around onshoring manufacturing. We'll have to see what the ultimate impact of that is, but also some proposals from FDA as part of the user fee process to provide a fee discount if folks bring their phase 1 trial to the US, as well as some proposals around streamlining the IND process, which I think we all can agree would be helpful. But to build on what David was saying, I don't think that it's the most productive, especially since there's so many unknowns, to sort of focus on blocking data from a certain country or region, and instead to think about how do we create an environment in the U.S. that helps to facilitate development, makes it a more receptive environment, so including funding, as well as a predictable and stable regulatory environment that can bring products forward, as well as advance regulatory science. I think that's just as important as thinking about how to block data coming from someone else. We should think about how to streamline INDs, certainly with patient safety as the ultimate test for INDs. But there's space that can be shortened.
So more of an approach of maybe using some lessons learned, but adapting our current system in order to address some of those pressure points that have been encountered. So in thinking about the impact of this geographic diversification and competition alongside of the scientific progress that has been accelerating various different forms of development, including in oncology. When you think about sort of the next 1 to 3 years of what will be coming front and center and what it's going to take to sustain a efficient and competitive environment in the U.S., where do you see that headed? Maybe David, I'll start with you. What does the venture capital and investment community need to see in order to keep things moving forward? Both globally, but even in the U.S.
Yeah, I think we need to very quickly reverse course on the degradation of our basic research infrastructure. And I think we need to take more than small step changes at a much faster pace than we're used to make our development ecosystem, our drug development ecosystem, more efficient here. And the way I think about it is, two things that are incredibly unique about our innovation ecosystem is one, it's relationship-based across biotech, academia, investment community. And yes, you can go globally to source innovation, but it's that incredible network of relationships that we have locally that is the sustaining force for the innovation, the new ideas. And the other feature is risk-taking. We've built an ecosystem where you can join biotechs that do very risky things. They can fail. You can go on to the next one. And you can build a very safe career, taking lots and lots of risks. And that's because of the nature of this overall infrastructure. So, when we have that, the dependencies are we need those great ideas, we source them through our relationships, and then we build them up because we have an efficient time cost to value structure. When things are more expensive, to actually get to those value inflection points of a pharma's interest or something else, then that makes the whole range of things that we can do much more narrow. We have to do much more conservative things. And then we've already just said, the pressure from China means that it's only the really innovative things that are going to be rewarded long term. So we got to fix this problem. And I think the imperatives are to fix the two things that I mentioned at the beginning in order to maintain the local level of innovation that's so important here.
Chris, maybe just to follow up on that a little bit, with uncertainties in funding, how do you think this has impacted, just given sort of I know this is a bit of speculation, but some of your past experience, how is this going to impact business at the NCI in terms of identifying priorities for funding?
I wish I knew the answer to that. I think the situation at NCI is terrible. I think we're losing an entire generation of mid-career experts from the NCI right now. And what you lose in that is not just someone who might invent a new technology. You're losing somebody who is a thought leader who shares those thoughts openly with everyone else in the academic community, and does so because it's part of their job to do that. And in losing that, we are going to lose a level of leadership in the clinical trial infrastructure. We're going to lose a lot of those people end up going to FDA as reviewers and leaders. A lot of those people end up on the Hill as advocates. It's awful. I don't know how else to say it. It's really terrible. Some of my closest friends who've been there for 20 plus years are leaving because they feel like they have no ability to do their job. Sorry, it bums me out a bit. But let me come back to the question that you, I think, are really getting to, which is, what is the ultimate limitation to innovation in this country? And that limitation is actually a lot of inefficiency that is created by spending money on things that are not necessary. So we talk about quality in clinical trials, but a lot of times quality comes down to a box-checking exercise that costs an enormous amount of money and actually does not generate more quality. And so why do we do that? It's because over time, that has become the standard process by which things are reviewed. But I think we need a really thoughtful look and understanding of what we're actually trying to accomplish and to clean that out as much as we can. We spend, as a community, drug development, we probably spend billions of unnecessary dollars a year on box-checking exercises.
And who could lead that effort? Is it going to take just a cross-collaborative moment in time to say, the system's under duress and we're going to need an overhaul here, or is there someone that can sort of step in and say, here's how we need to adjust?
Yeah, I mean, we live in a representative democracy, but the thing that is represented is money, right? So the problem is that too many people make too much money off of this inefficiency.
It's the clinical trials industrial complex, for sure.
Yeah. So, yeah, we would need our representatives in Congress to say this, enough is enough, and we need to get to the root of this problem and change the policy, because this isn't an FDA problem. FDA just enacts what the policy is, right? So this is a policy problem, but you need to take the time to understand what is really happening to fix the policy.
And does that stretch, this stretches just beyond sort of the drug development clinical trial process? This is encompassing legal,
I'm not going to
IRB.
comment on like financial issues, that's not my space, but I'm saying in drug development, we as drug developers know where those points are. And I think even FDA reviewers would say the same thing on their end. Like, we get way too much information that's unnecessary, but now that it's here, I have to read it all, right? And where did this come from? It came from some historic thing that just nobody has bothered to say, we don't need all of this anymore.
Yeah. And David, as you think about from like an investor sort of reviewing opportunities, is this just part of the current business paradigm that one must accept?
I think we don't control. We do everything we can in our individual businesses to build incredibly efficient development organizations, but ultimately we don't control these costs. And we need partnerships, and we need partnerships with folks like you guys who go out and try to influence policy. I come from Jake Auchincloss's district, and he's somebody who does think about these things and actually talks about these things, but I think there are too few politicians who actually do that.
Yeah. Julie, thoughts? How do we get congressional interest beyond Mr. Auchincloss to begin to examine some of these areas?
I mean, I think there is interest beyond his office, but obviously he's been pretty high profile about it and has a good understanding. I think I'd push back a little bit on the idea of Congress needing to reform the system, because a lot of this is in FDA regulations and guidances and processes that are implementing very broad stroke provisions. And so what I'd like to see in the coming years is, Congress really giving FDA direction about where to go, reopen the regulation on adequate and well-controlled trials, right? Reopen your IND regulations, do some workshopping, direct FDA to do more engagement, and make sure FDA is adequately resourced, both in terms of staff numbers and expertise to do that type of work, and to have the space to have some, not just meeting deadlines, but also taking a step back and thinking about where the pressure points are, where there's space to be more efficient, and sort of coming from the bottom up, in some ways I think can be really helpful.
And do you think there will be the opportunity to regain focus on these issues? You mentioned the user fee reauthorization. Is this a point in time, maybe not right now, but over the course of the next 10 months?
Yeah, I think the next 10, 15 months are going to be really ripe for conversations about what policy levers we need to put in place.
Well, maybe just to wrap things up, you've raised some heavy topics that we could probably go in depth on, maybe more appropriately not when we're wearing microphones around some of these issues, but just to sort of round things out, I'd love to hear from you guys, in thinking about over the next, 1 or 2 years, what are you most excited for? What's sort of on the horizon that you see as one of the most promising things that is coming down the pike? Maybe Julie, we'll start with you and just roll down the panel.
Yeah, I mean, I think that there's so much innovation happening that, and how to harness that. And I take a lot of, I know folks were talking earlier about, the platform technology provisions and leveraging prior knowledge, figuring out how to do that best so we can move forward innovation.
Thanks. David?
Yeah, there's so many places for optimism, and that's why I think we all do this work, is because science marches on and incredible things are happening. I think in oncology, I think we're going to see a next wave in IO. I think the in vivo CAR T approaches are going to get us to solid tumors. I think early intervention, early interception is going to be an exciting space.
Great.
Yeah, for me, I'm not convinced that AI is going to drive efficiency in clinical trials for some of the reasons David mentioned earlier. I think it is very much relationship driven in a lot of ways. Data entry mistakes, et cetera, happen because junior people are putting in really important information incorrectly sometimes. And I think there are ways with software that those things could improve. I'd like to see that happen. I think the other thing, just from a broad picture, where is AI and machine learning really helping us already? It's in protein engineering. We're seeing the ability for a very small group of people now to do the work that would used to take a year in a week in terms of three-dimensional modeling proteins, being able to understand their binding dynamics or interactions in a three-dimensional space, and that is just incredible. So once those tools are being implemented into some of the novel ideas that have been around now for 15 to 20 years, I think the pace of innovation of new drugs, even for what used to be called undruggable targets, is going to pick up rapidly. And what we really do need is an infrastructure to be able to support all of that.
Well, I want to thank you guys for spending a few minutes to share your perspectives. Obviously, to move things forward, each of the sectors that you're representing will be critically important, including all of those that were represented throughout the discussion today. So please join me in thanking Chris, David, Julie for a good discussion at lunch. I will let you guys off the hot seat and distract for a moment while we transition the stage into our next panel here. We're going to move to the third panel. If the session 3 panelists that hopefully have been mic'd at this point would start to make their way to the stage, that would be terrific. As we get started with a brief furniture rearrangement, maybe I will take a moment just to highlight a couple of upcoming events that may be of interest that we'll be working on over the next several weeks. We'll be hosting a series of online events, transitioning from our in-person meeting here today, to provide a series of new reports and results from several ongoing projects. These include several that are noted on the screen here. Number one, a forthcoming report on new ideas for optimizing the development of new biomarkers. We'll also be hosting an event on the presentation of new data simulations to help inform the interpretability of interim overall survival data in clinical trials at a time when an earlier endpoint is being analyzed. Third, a new report that's describing potential approaches for dose optimization during the development of new radiopharmaceuticals. And finally, our 19th annual meeting, which will be a hybrid event, the in-person of which will be here, hosted at the Ritz-Carlton on November 10th of this year. We hope that you'll be able to join us. That will also be simulcast on a webcast. So without further delay, I think we got our chairs switched. So our final session today will focus on longer-term policy and regulatory science opportunities to strengthen the cell therapy ecosystem, including areas that may benefit from structural alignment, pilot efforts, or legislative engagement to support sustained innovation and patient access. I'm happy to hand things over today to Lizzie Lawrence from STAT, who will be moderating this afternoon's discussion, and to a terrific panel to help guide us through these issues. Welcome. Thank you very much.
Thank you so much. I'm excited to get started. So just to briefly introduce everyone, I'm Lizzie Lawrence. I write about the FDA at STAT. We have joining us George Eastwood, Executive Director of the Emily Whitehead Foundation, Grace Graham, Deputy Commissioner for Policy, Legislation, and International Affairs at FDA, Preeti Hedge, Global Head of Research at Kite Pharma, Timothy Hunt, CEO of the Alliance for Regenerative Medicine, and Carl June, Director of the Center for Cellular Immunotherapies at the University of Pennsylvania. So there's a lot of people. There'll be a lot to say. I was saying I'll probably get like 3 questions in max. But to start, I thought we could just take stock of where we are now. We've seen some FDA flexibility on manufacturing, the plausible mechanism framework, CAR T label updates that enable outpatient delivery, and broader use of real-world evidence. So I want to ask, what you all make of the agency's current initiatives and where are the gaps? And maybe let's start with George, if you can talk about this from a patient perspective.
Sure. Yeah, and thanks for having us here. And I think starting from the patient side, it also starts from the story with Carl and Emily, where almost exactly 14 years to the day right now from when Emily was treated. And so much has changed. And I think we miss that. It's so different for patients. Something that I say quite often is, you know, the Whitehead family had this great opportunity to be in a 4 to 6 block radius of Penn with Dr. Grupp, Dr. June, Bruce Levine, all tackling these challenges together. And it's an unsustainable method to deliver at scale with the promise of the clinical side, right? So as time has evolved, the foundation has evolved to look at these issues too, right? It's not just about getting it to the first few patients. It's about getting it to the 80% that aren't getting it. And along the way, the foundation's evolved to try to solve these problems too. So we've really started to dig in and see how has it changed for patients? What does this relationship look like, and how do you scale it? So for us, it's asking, right? When you want to learn, you ask patients, what is different? What did you like? What did you not? So, you know, we started digging in on this long-term follow-up question, which is a very specific need. But when you dig in and ask patients, there's so much that's unknown about the process. And then when you ask Carl, he will say, let's treat more patients, these are safe. So the struggle in getting this work out there was publishing an academic paper, gathering the data, getting all this data together took about 1.5 years. And about 2 weeks ago, this was finally published. So is this the way that we need to enact change at the FDA, or can we have more direct dialogue? And can we get the patient voices in here earlier and more often? And not just for CAR-T, but for all these modalities. You mentioned plausible mechanism and others. How do we start the trials fast, but how do we get them approved clinically, commercially, and then deliver to all eligible patients? Those are the big questions we have.
Yeah, maybe I'll jump to Carl since you brought him up. You know, what would you say are the biggest opportunities for, you know, the biggest gaps in getting this to patients, and the biggest opportunities for filling those gaps?
Thanks, Lizzie. So first of all, I want to thank Forensic Cancer Research and the Parker Institute for doing this. And, you know, this is cancer research is what we're talking about today. It's not vaccines on healthy children. And the problem I see is that our regulatory strategies really don't differentiate enough between, you know, end-stage cancer or, you know, treatments and the regulatory approach for that versus, you know, not immediately lethal diseases. So, you know, we had, you know, that treatment that treated Emily back in, FDA approved in 2017. That was bilateral work between the University of Pennsylvania, Novartis, and the FDA to make something impossible. I mean, we never thought an individualized cell therapy would get commercialized. So it happened, and now there's over 50,000 patients treated. And we've learned a lot. You know, autologous cell therapies, you know, are safe. They rarely transform. And now we have something none of us saw coming, you know, autoimmune disease treated with CAR cells, in vivo injection of live viruses, you know, nanoparticles and things we didn't see 7 years ago. It's just an amazing advancement. But right now, our regulatory approach doesn't really distinguish adequately enough between, say, an autologous N of 1 therapy and versus in vivo delivery of a mass-produced therapy. So right now, most of the, I mean, we have this backup of science advancement where, but the ability as an, and I'm the only academic on this stage, but their early academic trials are very much under stress now, both from the funding aspects and then the regulatory burdens are much more difficult now, counterintuitively, than they were, you know, before FDA approval. So what's happened is academic trials now are more raised to a standard of like a registrational pathway. And I think we need actually to be like China and have an IIT-like single institution for early discovery, and then one that's a registrational, rigorous approach. And we just don't have that. The FDA needs to have a regulatory framework to be able to do that. And so that, I think, is, you know, we could talk a long time about how to do that, and then how, for instance, like autologous cells, I think, you know, should have different approach really than, you know, off-the-shelf things that are not N of 1.
Right. Grace is our FDA representative. I'll put you on the spot a little bit. How are you starting to think about early phase trials, trying to make those easier to initiate in the U.S.? I know that's something that's been a priority for the agency.
And thank you, and thanks to Friends for having me. It's been a great partnership over the years, both on Capitol Hill and now at the agency for a little over a year. But I think the agency has heard the message loud and clear and seen the data that early stage research is moving outside the United States, whether it's Australia or whether it's China predominantly. And a lot of our guidance regulations are both old and very broad in terms of not differentiating between a first in human, what you need, versus what you need for a later, larger stage trial. And obviously the public health risks and benefits may be different for those two, both the size of the patients that are going to get the treatment and obviously severity of the disease may necessitate different approaches at those stages. So there's a lot of active conversation. The commissioner has said sort of more to come in the next month or two, but it's something we look forward to having a robust stakeholder process. I think to the extent there are specific guidances, regulations that you think need to be improvement, have more flexibility. Another, I had the privilege, I know there's been a lot of great CBER folks here earlier today talking about this, but getting to work with them, learn from them. A lot of times we may have flexibility we use in the agency, but we haven't necessarily communicated that. And as long as something's in the IND stage, we can't really communicate about that. And so how can we start to think through ways to communicate the flexibilities we already use, and obviously plausible mechanism guidance that came out was a really cross FDA, CBER effort to put down some of those flexibilities I think the agency was already using, and for the first time signal that there is a path to approval for these N of one, N of a few diseases or conditions, where before I think folks weren't clear how to get those out of the IND stage. And I think it's really important the agency and we think through ways to get the good data we need to get that stamp of substantial evidence and traditional approval to facilitate broader patient access.
Right. And on the plausible mechanism pathway, I know the researchers behind the baby KJ treatment, they have had some difficulties as far as the manufacturing standards and trying to figure that out with FDA. How are you starting to think about whether it's possible to lower those standards for treatments in this plausible mechanism pipeline and make academics more able to produce these therapies?
I mean, I can jump in. I would much, the CBER expertise on the CMC or can definitely handle that question easier than I can, but I hope that is one area we can get comments on in the guidance, and as you saw with some level 2 guidances released this week around CMC, we can get feedback on those too, and I think it's helpful to differentiate sort of where the CMC flexibilities are needed, where broader patient populations are possible versus these sort of N of ones. And thinking more and more, we sort of had a traditional, you have a commercial IND or a research IND, and what happened, are some of the research ones now the only path to potentially commercializing some of these, and how should we think about those INDs and structures moving forward? We may have sort of non-traditional sponsors wanting to come in and sort of get approval for these things, and how does that work from a regulatory standpoint? How does that work if you have both academic sort of GMP level, but also you're trying to commercialize a product at the same university even? How would that work?
Mm-hmm. Right, yeah. I don't know if Carla or anyone else has thoughts on kind of the ways we could, the comments you'd want to submit to the agency on that, on how to deal with this, with the issue of manufacturing.
Look, I think the administration deserves a ton of credit for both the plausible mechanism of action concept and now trying to turn it into guidance, as well as the CMC efforts, right? We can talk about it later, but we're extremely high on this notion of using gene editing as a platform to kind of crank the flywheel faster as a good exhibit A on how to compete with China, right? And so I think thinking about CMC as really fit for purpose is what the administration is trying to do. Now, there's a comment period we've submitted comments, others have as well. That's the process, right? But to go from concept to draft guidance to then we'll get something, we'll get further finality on it all. I think there's a lot to like.
Yeah. So I want to talk about personnel changes at the FDA. There's been rapid changes in leadership over the course of the past year, a lot of institutional knowledge lost. And especially with acting positions in the CBER director and CDER director roles. Personnel's policy, and when the personnel keeps changing, it's hard for developers to keep up. I guess, sorry, Grace, I keep putting you on the spot, but Grace, how focused are you on trying to retain FDA as experts?
Yeah, thank you. It's a top priority. I think we know that we had a long time of a lot of great long-term center leadership between Dr. Shuren, Dr. Woodcock, Dr. Marks, long tenures. Anytime you have those sort of leaders leave, there's going to be some change and some turmoil, and kind of assessing that. I think obviously we had a lot of folks leave. The RIF was my third week, which is challenging to manage through, but the agency, I think, there's an opportunity where we have some great institutional knowledge still at the agency. We hope to retain and do all we can to retain and make it a great place to work, as well as then the opportunity to hire. The commissioner's made a priority of hiring and getting new folks in the door who may have more experience in some of these spaces that are sort of on the cutting edge, not that the scientists at FDA are not, but I think where you can have that blend of institutional knowledge with the new folks coming in the agency, I think it could create a lot of opportunities to look at things differently, do things differently, working through guidances, regulations, so you have that certainty, but it's a top priority to get kind of permanent leadership in, retain what we can, hire where we need to, so we can do our best to keep up with the fast pace of everyone in this room, just in the cancer field, and then obviously elsewhere.
Yeah. Anything you can share on who might be the next CBER?
No breaking news.
Don't touch it, Grace.
No breaking news today for me, but I appreciate the question.
That would be awesome if there was breaking news.
But I think from the patient side, it's the communication from that new CBER director to the patient. We've seen that in the past, and that not just the individual patients, but as we've expanded our mission into rare disease and gene therapy, it's the communication to these groups that has made the most sense. I spoke to 2 of my board members this morning who are in rare gene therapy, and it's just, what is the number one thing I can say on stage today? And we just need to know the map, the terrain, who to go to for what. It was that simple. It clouds our ability to get these direct asks together when we just need continuity and connection across the board to the advocacy organizations that work so closely with what we need to deliver.
Right. Yeah, I guess I'll ask Preeti or anyone else who wants to share how you felt about the FDA's personnel changes and how that's impacted your experience and what it means for developers.
Yeah, clinical development is a long game. It takes years and goes through multiple administrations sometimes to, from a start of a trial to registration. For us, predictability is critically important. Clarity and consistency is critically important so we can plan around it. You can be flexible, but if you're flexible on a use case by use case basis, it becomes really hard to plan for it. And from a sponsor perspective, when there is stability at the other end of the table, it actually creates more confidence in the conversations we're having, and then that allows us to pull through as the trial progresses to ultimately get to better outcomes for all. So critically important.
I would add, so there's a case for frustration and a case for optimism, right? So within our community, people are very frustrated, and within the rare disease community in general, right, with the number of complete response letters that we've seen in the last year and a quarter or so, right? I think it's 5 in the last 15 months versus 5 over the previous 5 years, right? I have a son with a rare disorder. We have over 50 patient organizations within the Alliance for Regenerative Medicine patient organizations. Many of them are focused on rare disorders that you've never heard of, and it would break your heart if you hear their stories. So the frustration around using complete response letters as a tool and also pushing randomized trials in areas that a lot of people in the patient and physician community think are impractical at best or unethical potentially. The frustration is very intense, right? On the one hand. On the other hand, to be in biotech, you have to be an incurable optimist, as somebody, I think, said earlier today. And I do think there is a big opportunity for a reset within CBER and OTP that is needed and can be very helpful and incredibly constructive. I would say on regulatory flexibility, that's a great example. almost by definition, that does not come bottom up. It has to come top down from leadership. You can't hire reviewers and say, good luck, you're a year on the job, show regulatory flexibility, right? It's not practical, right? It really has to be leadership down. And so I think that's a good example where the new head of CBER can come in and say, look, I do have a different mindset. My mindset is we want to find the problems early. Don't be a problem identifier. Yeah, identify the problems, but then push to resolve them favorably in a timely manner, working with the sponsor. And if there's reasonable areas of scientific disagreement, find off-ramps like advisory committee meetings or type A or type B meetings, right? Because the big mission, I think, is shared by the great professionals at the FDA. We've heard from many of them today, which is we want to see these medicines get into the hands of patients so they have choices. No one wants a gene therapy or a gene editing medicine because they're healthy, right? They're on the clock. Time is life and time is functionality. So I think there is a case for, a refresh that could be quite promising.
I'll give you an example of the conversation that was had this morning around MRD as well, right? It's a biomarker of disease at the end of the day. It's not exactly a therapy-based biomarker, but biomarker of disease. If sponsors have started registrational trials with MRD as an endpoint 2 years ago, and it's now not considered an endpoint, it's worth having that discussion in a forum that allows us to say, how do we now adapt to it? Just those fundamental questions of how do you look at a biomarker for a disease, specific disease versus a modality, I think is important as you're running trials.
Yeah, absolutely. I'm curious, what you guys would like to see from the FDA as far as, I know there have been some kind of listening sessions with industry. There have been fewer advisory committees this year, but so are there more opportunities for the agency to engage with different stakeholders? From anyone, yeah.
From the FDA,
yeah.
or would you like me to comment?
Yeah, you can start, on the best ways that how
Sure,
the
I think
agency
there
would
was
like to
recently
interact.
an example on cell and gene. CBER hosted a workshop with, I think, ASGCT and ARM and a bunch of folks.
ASGCT,
ARM, sorry, Tim.
That's right. That's right.
And I wasn't able to make it, clearly. But, I think those are really useful so you can have these conversations about endpoints and the best ways to do them outside of a specific product context. And so hopefully continued work like that can continue, and FDA will continue working on those, as well as I think advisory committees are valuable, and getting the right expertise on those is important, and using those to sort of have some of these decisions where the science is a tough call and the risk benefit may be a tough call, and making sure there's a way for the patient voice to be heard, as well as sort of very frank and open discussions about the underlying data.
Yeah, and having been a part of that with Tim and ARM, one of the recurring themes was the inability to get children into trials first and this need to go into adults. And I think we were continually met with the story of that's not what the guidance says, but it was every single person. I know Nirali is here. She was on one of the panels. And it was the same thing over and over where the guidance says one thing, but in practice, every single person on every panel said, well, we're not getting that answer. And I think it speaks to what Tim's talking about, which is that top-down interpretation. The guidelines are great, but how do you train on them? How do you interpret them? And then how do you make sure that that connection happens? I know it's incredibly hard. I came from the CMC side of cell therapy. So the joke earlier about the 18-month difference in the choo-choo train, I lived that in CMC versus clinical, so it's not easy at the FDA. But how do you be the one to connect those dots a bit more for the sponsors that are moving fast and quickly? I think that's something the FDA could do to help out a lot.
Yeah.
Lizzie? So, one of the issues I think, there's been this growth of just bureaucracy. So part of the problem is we are the problem. It's been much harder at my university to get a material transfer agreement approved than it used to be. We've had huge growth that way in bureaucracy, and that's happened throughout. And China doesn't have that because they basically engineered a system and didn't have all the legacy. So theirs is a more engineered built system. I think What's going to face the FDA is hundreds and hundreds of INDs with cell therapies, and it's not going to be just cancer, it's going to be regenerative medicine, autoimmunity, and that's a huge problem that they're facing and on the horizon. One thing you can look at is what do they not need to do, and I think for early stage autologous T cells, the FDA does not need to regulate those. In China, you don't go to their FDA for the first test. You check it with an IRB approval, and we don't have the ability to do that. I think we should have single institution, test academically, and then if something looks good, based on your local IRB, this is not for in vivo delivery of in vivo delivery of, for instance, a viral vector, but for an autologous ex vivo engineered cell, I think that should be like bone marrow transplants. Those are autologous cells, but they're done with IRB approval, and that's they're N of 1 therapies. If it passed that test, then I think it needs full IND approvals and to enter that pathway. But that would decrease FDA's workload a lot and speed this initial discovery effort, which is what I think we all need to get to, in the case of solid cancers.
And I'll just jump in. I think ideas like that are welcome, and the agency is trying to do creative thinking about that and welcomes that dialogue. We put forward in our budget a legislative proposal to sort of look at expediting the IND in certain ways. Obviously, Australia sort of has a similar system, but for more lower risk, not cell and gene therapy. China, I don't know if their IITs do cell and gene therapy. You're probably the expert. They do, right? And so, that is also, the way our statute is written, anything that's going to be moved in interstate commerce is going to have an IND that would require working with Congress. But as the previous panel talked about, I think between what we're seeing out of those other countries, the need to maintain FDA's gold standard, protect patients, but being able and having experts say there's an ability to do that with the potentially new models, the administration welcomes that dialogue and being part of that conversation with Congress and patients.
Yeah.
Can I just add one more thing to what Carl mentioned, and this is around platform iterations, and I'll just use in vivo cell therapies as an example where, say, the CAR T has been studied in 30,000 patients, okay, through an ex vivo autologous platform. But you've got an in vivo system that you would like to iterate to understand how best to deliver it safely and effectively. Today, we don't have a pathway that's efficient enough to do that in the United States. Just as an example, in China, you can go from changing the targeting binder to a patient in 5 months. It would take us 5 years and a lot of time and resources to do it here. So that platform iteration piece where, say, your CAR T is the same, you're just iterating the viral vector platform and manufacturing. How do we do that more nimbly and efficiently in our ecosystem is my question. Today, we just don't have a path to doing that, and that's where we lose the competitive edge.
Right.
Yeah, maybe just to step back for a second. If you think about it, the administration has a very compelling vision on where they want to go with cell and gene therapy, right? They've articulated that a lot. Especially last year, right? They want to be the global leader. They want to be faster into trials. They want to compete with China, right? They want to get things into the hands of patients, and they've articulated sort of going back, I don't know, probably in the first half of last year, like, look, we believe in the right and the spirit of right to try, right? Check, 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 check. All, I think, incredibly compelling. And Carl and I were at this last June when they had there were about 20 of us that were convened up at the FDA for a workshop on cell and gene therapy, and everybody can kind of talk about where they see things going and what advice they'd give to the FDA. And there was a huge listening tour that the FDA did. I mean, again, check, right? Like, these are really important engagements that I think represent the best of what the agency can do by convening and then leaning on others to convene this pediatric cell and gene therapy pediatric clinical trials workshop. The FDA called us up and they've been talking to us about it for 2 years. A few people left, and they said, we still would really like to do this. Can you help us stand this up? And the feedback's been really positive. It was patient organizations, prominent academics, industry, regulators, right? So I think the power of the FDA to convene or lean on others, and I give Jeff and the team at Friends credit, this has been a really good convening. I think the more the administration and the new head of CBER can get out and lean in and do those things, what they will find is this is a community that wants one thing, to help them execute their very powerful vision, to take the vision and help it become a reality. And then tens of thousands and hundreds of thousands of patients' lives, I firmly believe, will be dramatically improved and saved.
Yeah.
So more convening. That's what I'm selling.
Well, speaking of that, I want to ask you all, what can be achieved during the upcoming user feed discussions? What are there specific areas where additional resources, programs, or authorities could meaningfully help with efficiency and predictability in this space? And maybe Grace will start with you about, what's realistic and what's not.
Sure, happy to start. And obviously the meeting minutes are public for the agreements themselves, and look forward to working through the process where those agreements become their public meetings on the proposed recommendations once we have those with industry, with the patient input that we've been getting all along the way at the monthly meetings, and really looking forward to hearing the thoughts on those, and the goal then is we transmit those recommendations to Congress by early next year. And then Congress has until September 30th of next year to pass it, because it is considered must pass. From my time on the Hill, a lot of other FDA policy changes tend to ride along with it, or shortly thereafter, as it happened in 2022. And so there's a lot of opportunities, I think, not just within the agreements themselves to look at, FDA has never met the meeting goals the way they should. And I know I was personally surprised at the pre-check meeting on domestic manufacturing, how much time was talked about scheduling, and how scheduling of inspections can be such a barrier or a challenge, especially in a space like in this one where you need to be making it live, but if you're making it for 8 patients, how do we sort of sort that out? But I do think there's some opportunities there through the user fees to look at some of those processes where earlier engagement can be helpful. I think in terms of resources, there's a lot going on at the agency to try to modernize on the tech side and AI and ELSA, and a lot of the summarizing the data, pulling perhaps some of the meeting management, other things, those tools can be useful in helping with as well. And then as I mentioned, as part of the budget process, if you look at that beautiful 200-page document on some time when you need to go to sleep, but there is about somewhere in the 20-page mark is a list of legislative proposals the agency has put forward, administration has put forward, a number of them, and would welcome, those are places I think the agency thinks is ripe and recommends Congress take a look at, and we would welcome working with them and, again, stakeholders on changes in authority. And obviously there'll be another budget next year, and so there may be additional or new things the agency put forward then as well.
From our side, on the patient side, we took part in the stakeholder listening for the PDUFA, and something really struck me. Somebody mentioned ELSA, I won't say, but in the meeting there was a comment, yeah, we have ELSA, but nobody needs help writing an email. I beg to differ that communication is important in these things. And at the foundation, we've been heavy AI users, but in a very practical way, agreeing to do what we say we're going to do, recording meetings, having follow-up. But I think it starts with that practical use. I love all of the innovation. I came from that world. Trial matching, target discovery, you name it, that will come. But I think if we take a more practical look at the technology we have and what we can deliver today, that's something. But in those meetings, I think when we dismiss the practical use, like reviewing a 1,000-page document, I think there's a lot of work that can be done that just needs buy-in and people to understand what AI means, what it does, and where it can actually save time in the minutia. And then we can apply it in a very trusted way for patients and in a regulatory manner in the future.
I'm curious what you all, what you'd like to see come out of the user fee discussions, or, the accompanying legislation that may go with it.
I'll pass it on. I'm not qualified.
Go ahead, Tim. Yeah, look, I think there's resourcing issues and there's structural issues, right? I think we've been very focused on the structural issues. But I think on the resourcing side, yeah, there are opportunities. The FDA hired a number of, over 100, I think, reviewers with an OTP a few years ago, right? They're still fairly early in the arc of their career. For many of them, they'll stay for a little while, then in the natural order of things, they'll transition out, they'll go work for companies or whatever, right? So I think there's a continuous need to hire people and train people. And that's always been the case, right? This isn't something new in the last year and a half. And I think having that mindset of continuously thinking about we have to hire and train and it's never ending, I think is probably one of the areas I would argue would be resources well spent.
Yeah. I wanted to ask you also about, you're talking about a top-down approach. There has been, a lot of controversy lately, and in general, whenever this happens, when kind of a center director overrules staff, or there's a perception that there's been, politicization of the process. I guess you have concerns about that. How can you see career staff starting to, align with leadership? How kind of important is that as far as, making sure that there's trust in the process? It's kind of a...
Who wants a piece of that first?
I mean, I think, we need this to be science-driven. Yeah. And that I think is the way that it needs to go forward. And I mean, I've seen other healthcare systems where, it's very much made, decisions made based on policies and not science. And so we need to really, hold the standard that science is what's going to guide our regulatory decisions. And it's, the problem is, it's one of these things, good news, bad news. I mean, the science is moving so fast in cell and gene therapy that the scientists can't keep up, and it's so hard for the regulatory scientists to know. I mean, so we had this meeting 2 years ago that Friends of Cancer organized, because there we were trying to basically educate the FDA on the idea of phase 0 discovery in humans, where we have trying to just find out which targets are the best. And you can do that in CDER and where you inject several compounds and find out which one, but we're not allowed to do that in CBER, phase 0 discovery things. So we had, a very nice, back in 2 years ago, almost to the date, about this, socializing that with the FDA. And because they didn't, the things have gone so fast from CRISPR to base editors, split base editors now, and in vivo editing. So the FDA, to keep up with that is near impossible. So they need, huge, hiring to be able to keep up with this. But that's, the problem is they haven't been able to keep up with that. And we need, I think, a pathway that will allow early stage, basically academic drug discovery in cell and gene therapies. And then what we already have in place is a very good system, I think, for commercial development, other than, I think, the issue we haven't talked about, which is something, Tim, orphan drug development is another huge thing that we haven't talked about today.
Yep. I would say I do think it's a tricky balance, and the extremes are incredibly unhelpful. What do I mean by that? Nobody would want head of CBER or senior CBER person or a head of OTP to be, weighing in on all these approvals, right? That's crazy. That's the lack of management, right? It's micromanagement, and no one would want that. On the other hand, I do not fundamentally believe, and this is not a near-term thing, this is a many-year thing, you can just put review teams on autopilot and say, have at it. Because again, you might draw, depending on who's your reviewer, you might draw someone that's really junior and that is incredibly risk adverse and that's going to hug the playbook so narrowly that it's highly problematic. And that's where, guess what? In any organization, you need leadership, management, people that can make judgment calls, and that has to come. And that can, as I said earlier, exercise judgment around regulatory flexibility, and that has to come top down. That's management. And you know what? Sometimes you catch a bunch of arrows for doing it. Oh, fair enough. But you can't do it, shooting from the hip. You have to do the homework and really kind of look at this. But I do think the best path is to get the review teams together early and say that we know there are going to be problems. What are the problems, and how are we going to resolve them favorably? And that's leadership. And in the absence of that, it's a huge frickin' problem because it looks like things are on autopilot and no one's in charge.
Right, right. Yeah, I don't know, Grace, how are you thinking about kind of, because obviously people will have disagreements, but trying to achieve that kind of consistency to minimize situations where there's a sudden overruling or switching of decisions.
I think, from my career on the Hill, I think it was the most routine complaint at my door was someone feeling like, hey, look at oncology, they're using accelerated approval way more than another center office is using this tool, or CBER is doing something in a different way CDER is doing. If everyone could just behave like oncology, it would look very different. Obviously, there are going to be some cases where, whether it's the science behind the product or the nature of the product itself and the risk of the product, the risk benefit of the population being treated, that there are those differences. I think the goal, again, is to be consistent and avoid surprises, and where things start to look like things may be changing, or the data doesn't pan out the way that folks discussed, or if a company chooses to do something different than FDA may have advised, that it's clear when they submit why they did that and why they think it's sufficient, and having those conversations in a constructive manner is sort of important in the goal.
Right, right.
And I think from our side, across what everybody has mentioned, Carl mentioned pointing to the science, Tim mentioned the structure, but I think from a patient empowerment side, how do we take into account the full picture that's beyond the science and the structure? Speaking to, I mentioned a rare disease board member of mine, there is a perception piece of not wanting to treat a healthy patient first, but in this neurodegenerative space, we know eventually what's going to happen. So these delays that happen, you're thinking about some negative publicity that might happen, but the parents and the patients themselves know what the outcome will be. So how can we deliver to them faster and let them make that choice? Sure, you may cause something disastrous, and this is from an earlier conversation today, but we know the last 5 years is going to be suffering for both the patient, the parent. So how do we empower the patients to make those choices while still honoring the right pathways that we're talking about and the science? Because there's a very human component that we don't measure well in what do these years look like, especially in rare disease.
Right. So I'm going to ask one more question, and we're going to do some Q&A. If you're thinking about what's achievable in the next 2 to 3 years through existing authority, PDUFA, or legislation, what are the 1 or 2 changes or opportunities that would have the greatest impact on how these therapies are developed and reach patients? I know we've kind of been talking about that this whole time, but really briefly, if we just want to go down the line, like what are the 1 or 2 things you think would be the best things to do?
Yeah, and I think you can zoom all the way in on something small and then zoom all the way out. And I think maybe the question we'll have next points to some of this. It's how do we deliver what we have today? This paper that Carl and a lot of the commercial CAR T companies and the Emily Whitehead Foundation and Blood Cancer United combined on is looking at the safety data and cutting long-term follow-up for commercially available CAR T from 15 years to 5. It will, allow a lot less burden on the provider, on the patient. So it's a very specific piece that takes a couple years of digging into. But then I think when you zoom all the way out, it gets into this use of modernization and tools. Within that same survey that kicked off the long-term follow-up work 2 years ago, we started asking, are patients comfortable sharing their health records with their sponsor? How comfortable are you with AI? And how about we ask patients how they look at these technologies, and we can integrate it into the modernization plan for the really long term. And in convening with the FDA, how do we bring that patient voice together?
From my perspective, really excited about the IND reforms. Obviously, Carl talked about what one model looks like, but I think there's a lot of options and a lot of work the agency can do on its own, plus having conversation around authorities, I think can unleash a lot of it earlier to get to patients earlier in the United States, and then hopefully the clarity around plausible mechanism and other pathways can get that eventually to patients through traditional approval and that substantial evidence standard sooner.
A thing that we didn't talk about this afternoon, but 80% of the patients are seen in the community hospitals. And CAR-Ts today are still administered primarily in the academic institutions. So what can we do collectively as a community to get cell therapy access in the community hospitals, whether it's through reimbursement policies, whether it's through using real-world data to show that community hospitals can very safely and effectively administer cell therapies, I think is going to be important, because then you can get more patients onto clinical trials. We're limited by the number of patients that can get onto clinical trials today, because there are only this many centers that can administer these therapies.
Yeah, that's a good one. I think that's a good area for convening. To keep harping on my topic, 80% of the patients that are CAR-T eligible are not on therapy. The FDA label change that took place last summer and removing the REMS is a big step forward to some common sense reforms. I think convening on that topic would be a great idea. Look, I just think that there are a lot of really good guidances that are in the works. That was, I think, it took the administration a little while to get its sea legs, to get that kind of thing going, the machinery going. Pulling a lot of those through, I think, could be really meaningful. I mentioned earlier the plausible mechanism of action concept and how that might enable gene editing as a platform. If you're a small company, we talk about like the challenges with rare diseases, and there aren't enough companies working on them, but if you could actually accelerate that through platform technologies, you could think about it not just in the US, but with the MHRA and with the EMA, and we're in touch with those jurisdictions as well. So suddenly you could take a patient population that's very small, and you're now looking at 3 geographies where it could be much bigger, and the economics make sense to put shoulder to the wheel. So that's one example of many things that I think are going on that could go very well through the guidance process. More convening, more guidances to enable the field.
Okay. My takeaway would be this dual track system, that the FDA be given a way to have IIT trials, whereas they don't at this point. And then that would, I think, both accelerate the discovery and development of new cell and gene therapies and make it more rapid and cost effective.
Awesome. So our first question, let me see. So what is being done to reduce barriers that delay access to potentially life-saving cell therapies, especially when timing is critical for patients with aggressive or relapsed cancers? Who wants to take that one?
I mean, one is by democratizing access, by getting cell therapies to community hospitals. That does take engaging on what does FACT accreditation need to look like for cell therapies versus BMTs. Today, our guidelines are still fairly BMT-centric. What needs to be done still is simplifying the reimbursement process. Patients can't wait months to know if their cell therapy is going to be reimbursed. These are very aggressive diseases. They progress really fast. So what can we do to make that reimbursement process much more simpler for patients and their caregivers is going to be important. We're doing a lot from a manufacturing consistency perspective, turnaround time perspective, to make it more predictable for the treating physicians and the patients. We're still developing next generation cell therapies to further improve on the transformative benefit, make these cell therapies more safer. So there is a lot going on the science and R&D side to make this more accessible from a safety perspective as well.
Maybe if I could just add on that topic, I mean, that's one that I don't think has gotten the attention it really deserves, because, we talked about this 80% of the patients can't get access to the treatments, right? Well, why? Right? There's a number of confounding factors, but one of them was the labels for all the CAR-T companies went back to 2017 and had not been updated since then. And I was at 2 meetings, someone on this panel was here with me, where we were talking with the administration, and the folks said, look, we want to accelerate things and move things along and reform things and restructure them. And so we were encouraged to submit some very specific ideas. And we took the time and we submitted, I think it was like 8 of them in May of last year, so about a year ago. This was one of them. And we were like, look, it's kind of crazy, because after you get dosed with CAR-T, you can't drive for 8 weeks. And you had to spend 4 weeks within 2 hours of a major treatment center, essentially where you got dosed, right? Well, that probably made a ton of sense back in 2015, 16, and 17 when the data was being collected, and you would know better than me, as would you. But did it make sense now? It looked like what the data suggested was most of the serious side effects take place in the first week, maybe the first 2 weeks, right? And so the FDA, I think, did a nice job and kind of cut those requirements back dramatically to try to what? Enable CAR T therapies to get into the community. And so I think that's a great example of having a vision of where you want to go with cell and gene therapy and doing some very specific things to kind of try to drive greater access.
And I think from our point on the patient side, I spent last week, I think Tim was in Rome with the Alliance for Regenerative Medicine meeting. I was in Orlando with the Community Oncology Alliance, and the excitement around CAR T and their want to take this on is palpable. And it really does tie into the REMS changes, the long-term follow-up changes, and these little pieces that allow them to bring it in, because there's also the financial side. So I think it's really hard to not look at that whole picture and see that it's all patient-related. Right, these REMS requirements sound like a regulatory piece, but it also shows that it's safer for the patient and their confidence grows. And it's our job to communicate that to patients. But the things they're doing in the community, I moderated a panel on phase 1 clinical trials in community physicians' offices, which I knew nothing about. That's why I agreed to moderate. I would not add much to the panel, so let me learn and ask questions. But it was mind-blowing that is even possible when we think about what Dr. June is talking about. But there are community hospitals starting bispecific phase 1 trials. So I think we're missing out on how advanced community oncology is and how much better the CAR T treatments have gotten, and how do we converge to deliver together.
We're starting to see a shift for CAR T and other cell therapies where they're beginning to be developed in earlier disease settings rather than only R&R. What are the implications and concerns for patients as trial designs change to reflect this shift?
Well, I'll say, without getting over my skis, I mean, one issue is, MRD measurements and how that's allowed to guide approvals, so that's one issue. I think where we're really going to have an issue here is autoimmune disease. I mean, there are many more patients with autoimmune disease. We're seeing amazing results, in many trials. The FDA is going to be hit with this, and then rheumatology and how is it implemented in medicine, and when do you, it's easier in cancer where we have to treat patients up front, but the autoimmune disease thing is just the beginning of a tsunami of issues like this. So I think we're going to see upfront cell therapies. Those trials are underway in myeloma. I mean, there was an amazing AACR abstract last month in people with smoldering myeloma. And there were 30 patients treated, and 30 out of 30 were MRD negative afterwards. So there's a disease where only some of them go on to get advanced, but that's cancer interception. And that's where we're heading, but then there's an issue, how do you pay for it, and so on.
Just to add to that, I think an area that could benefit from a Friends of Cancer like forum is defining unmet need in early disease. There are curative therapies in early disease, and some patients may not need to go on intense therapies as well, but can we come together on defining what that definition of a high-risk patient is, whether it's in smoldering myeloma, whether it's in frontline lymphoma. I think that would be helpful for patients as well as for treaters to say, yes, this is the high-risk patient that requires to go on X and Y therapy. The earlier line therapy spaces are fairly crowded, but the more unified definitions we can identify, I think it serves everyone well.
Yeah, I think to Dr. June's point, in autoimmune, Tim invited it to arm the first MS patient, Marcy, treated with CAR T, and I was fortunate enough to have a conversation with her, and it really does change the way we look at things. She refused to take any immunosuppressives, but when we sat and talked, her question to me was, if you knew you were going to die in a motorcycle crash, would you ever drive a motorcycle again? So I have an immune deficiency, and I'm going to take something that's going to suppress my immune system. No, thank you, I'll wait. And she waited, and this earlier line was, I'll do CAR T, and she's doing great now. So I think that ties into patient empowerment, how we let people seek the treatment they want. But it really was eye-opening listening to her in that explanation. I mean, I'm personally afraid of motorcycles, and I would never drive one, but it really is how patients look at this.
Dr. June, you mentioned the complexities introduced by individual institutions, IRBs, tech transfer. Are institutions at a point where there is a recognition that this is a barrier, and is there willingness to change?
Sure. So at our institution, our hospital system, over the last decade, there's been a 3,000% increase in administrative positions and only 130% growth in the physicians. So we have, this massive bureaucracy that's happening, as that's one issue. But the real issue is also indemnification. And our institution would be much willing to do earlier stage trials if there were not class action lawsuits. In Germany, they have this individual health authorization ability, and there aren't class action lawsuits. And we need a way to limit that here, I think, to encourage so that these earlier stage treatments can get tried without, right now, the worry of litigation. I mean, that's I think, a legislative change that would really massively change if we could get that enacted.
And one thing I'd add is I have a dual hat at HHS as well, and I will just say this is something all of HHS is looking at, and so obviously NIH may have different levers in the FDA working closely with academic centers. NCI, Dr. Latai has talked a lot about this and been involved. There's also issues a bunch of patients have written to about sort of starker anti-kickback concerns about enrollment. I mean, there's a number of places that HHS can look at, and if we can remove friction, perceived or real, I think that is a goal and an effort that HHS is working on.
Yeah, amazing. I think that did anyone else want to add anything? Or? I think that's our last question. Thank you all so much for staying to the end of the conference, and thank you all for joining. This was a great conversation. I learned a lot as a moderator, so.
Thank you very much. Really appreciate everyone's time and insights. Thank you, Lizzie, for a great job moderating. I really want to thank all of the panelists from our earlier sessions as well as the working group that put the time in to help develop the issue brief to guide today's discussion, and certainly to our colleagues at the Parker Institute for Cancer Immunotherapy for their continued partnership and helping to facilitate these types of venues. As part of our ongoing educational series on June 11th at 1 p.m., we'll be hosting an advocates webinar as a follow-up to some of the aspects that were raised today during the discussions, and we hope that you'll be able to join us for that event, as well as our other events scheduled throughout 2026, which can be found on our event webpage. So thank you all for joining us today. We look forward to seeing you soon. Safe travels. Thank you.